हेलो हेलो हेलो। पहले वाला वाला टेस्टिंग वाला या अभी वाला?
अभी वाला।
अभी वाला हेलो आया।
क्योंकि आपका माइक ही नहीं था ना, तो कैसे आवाज जाएगा? अभी वाला माइक बंद कर दो।
A very good morning to honorable A very good morning to honorable dignitaries, down delegates, respected teachers and participants. I, Dr. Shashikala Prajapati, from Department of Zoology on behalf of Giri Society, Science and Commerce College, Kalamboli, feel privileged to welcome you all for this one-day National E-Conference on Frontiers in Biological Sciences. As Kurt Lewin once said, no research without action, no action without research. He was a German American social psychologist. He is known for his theory that human behavior is a function of our psychological environment. Before introduction to our conference, let's begin with the university song. Thank you for playing the university song. Conference can be a great opportunity to have an intensified exchange and debate on particular research issues of your choice. So this national conference will give an opportunity to exchange our ideas with each other, and occasions like this help us to test our knowledge and understanding. We are looking forward to get an exposure about what the best of the brain thinks over the dynamic issues of frontiers in biological sciences. This national conference is organized to provide a platform for industry experts, researchers, and students to effectively exchange ideas and recent advances in the various fields of biological sciences. It also provides a stage for budding students, researchers to share their findings and ideas with global experts. Life science is originally a biological science, but today it has matured into a form of integrative science with technological advances in molecular biology and biotechnology, which has led to burgeoning of specializations and new interdisciplinary fields like genetics, systems biology, molecular biophysics, nanobiotechnology, bioinformatics, etc. Now I would like to introduce the advisory committee members. We have Dr. R.J. Verma, who is ex-professor and head department of zoology, University of School Sciences, Gujarat University, Ahmedabad. We have second advisory board, Dr. R.V. Devkar, sir, assistant professor, Department of Zoology, Faculty of Science, the Maharaja University of Vadodara. Now I request our Honorable Principal, Dr. Giri, to convey a few words for today's national conference. Over to you, sir.
Happy morning, all of you. Today we are gathered here on the occasion of online national level conference on the topic Frontiers in Biological Science, which is organized by the Department of Zoology, KLU College of Science and Commerce, Kalamboli, Navi Mumbai. First of all, I welcome Dr. D.D. Jhala sir, Professor, Head of the Department, Zoology Department from Gujarat University, and today's session chairperson, Dr. Savitri, Katlam, MGM College, Mumbai. It is an opportunity for all of us to see, to look into the subject which is important in these days. Earlier, we used to live in the nature. We ourselves, with our deeds or misdeeds, we have shifted ourselves to industrialization. Again, we have to go to the nature. That is because the several problems, environmental, ecological, medical problems to which we are coming across, we are facing, there is a need to address that particular problem in a right way, and biology is at the top, which needs a proper concern from the people of all level of fields. Such type of national conferences guide our research students, encourage our researchers, and make aware the industry about the ecological changes. I am happy that several numbers of papers are presented by researchers in this conference, and the conference will be very successful. I am sure that there will be deliberations on the different aspects of biological science. And today it is an opportunity for all of us to listen to Dr. D.D. Jhala sir. All the way, he has spared his time for this national conference. I welcome Dr. Jhala sir, Dr. Savitri Katlam madam, all researchers, all participants from the different corners of the country. On behalf of KLE Society, Belgavi, and also on behalf of our chairman, Sir Dr. Prabhakar Kore, our KLE Society is 107-year-old society having 293 schools and colleges, not only in India, but in different corners of the world. Once again, I welcome all of you, and I'm sure that we'll be enlightened on this important aspect through the deliberations and discussions in this whole day. Thank you very much.
Thank you so much, sir, for the words of wisdom. I would request Associate Professor Dr. Usha Karunakaran, Head of Zoology Department and Convener of today's National E-Conference, to introduce our today's keynote speaker, Dr. D.D. Jhala sir.
Happy morning to all. On behalf of the KLE Society's management, Principal Dr. Giri, faculty members and students present for the one-day National E-Conference on Frontiers in Biological Sciences. I, Dr. Usha Karunakaran, extend a warm welcome to the keynote speaker, Dr. Devendra Singh Jhala, as well as the advisory committee members, Dr. R.V. Devkar and Dr. R.J. Verma, as well as the session chairperson, Dr. Savitri Katlam. It gives me immense pleasure to introduce our keynote speaker, Dr. Devendra Singh Jhala, Associate Professor, Department of Zoology, BMT and HG, Gujarat University, Ahmedabad. He has done his graduation from the Gujarat Science College, Gujarat University, and he's also achieved a gold medal in BSc Zoology, whereas he's completed his master's degree as well from in the area of cell biology and cytogenetics. He further went ahead and did his MPhil in toxicology and cytogenetics. He has completed his PhD degree in the area of toxicology and human cytogenetics from the Department of Zoology, University School of Science, Gujarat University. Now, as regards his academic appointments, he has been an assistant professor from the year 2005 to 2017, and from 2017 further ahead down the line. He has been serving as an associate professor at the Department of Zoology, University School of Sciences, Gujarat University. Now, coming to the areas of his research interest is very, very wide, which includes the genetic etiology beyond major health hazards, population genetic screening for mutation and integrating the results with susceptibility, evaluation of environmental elements for its mutational potential leading to genotoxicity or cancer, scanning herbal products as candidature for ameliorating property against mutagens and against metastasis, as well as genetics of Down syndrome, idiopathic mental retardation, ambiguous genitalia, amenorrhea, spontaneous abortion, infertility, endometrial implantation, and the list goes really a very long list it is. Besides this, he has received several research grants or fundings for major research projects, again, which is a very, very long list, and it's including a really high amount of funding, which is really a matter of great pride. Regarding the memberships of professional societies, here again, sir has a very, very long list. Some of them I'll mention. He's the fellow of the Society of Earth Sciences India, he's associate fellow of Gujarat Science Academy, life member of the Indian Society of Human Genetics, life member of Society for Reproductive Biology and Comparative Endocrinology, life member of the Indian Society for the Study of Reproduction and Fertility, life member of Biotech Research Society of India, is the founder director of Association of Zoology, life member of Bird Conservation Society, member of Association of PhD in Medical Sciences. He has received several awards and distinctions. As regards his professional services, he has basically worked as a subject expert in various areas. Main is the 5 e-content program for NME ICT project and for the website of MHRD Government of India. He has served as a script writer and subject expert to develop the e-content modules for MOOC for Swayam platform of MHRD New Delhi. He's a recognized PhD guide for the subject of zoology, biomedical technology, human genetics, life sciences, and wildlife biology and conservation. He serves as an advisory board committee member as well as evaluating the quality of performance as an NABL internal auditor. Besides this, he has served in several positions with respect to administrative activities, the list being again several pages to his credit. He has attended several workshops, both national as well as international conferences, symposiums, which are the number going to above 70. He has published about 43 papers and 108. So he has presented his papers in 108 places as in symposiums. He has, besides his research guidance, he has guided more than 30 MSc and MPhil students for their dissertation in zoology, biomedical technology, and human genetics subjects. He has guided 7 PhD students for their research work and 4 research scholars awarded PhD degree after successfully completing their viva. Regarding his citation indices in Google Scholar, he's almost having 624, whereas the H index is 15, whereas the I10 index is 22. Once again, I extend a warm welcome to Dr. Devendra Singh Jhala. Thank you.
Thank you so much, ma'am, for introducing our today's keynote speaker. Now, I request our honorable keynote speaker, Dr. Didi Jhala, to address the audience. Over to you, sir.
Professor in the Department of Zoology, Biomedical Technology and Human Genetics, Gujarat University. Society, Ethics, Science and Commerce College, Patient, Department of Zoology and IQAC, on frontiers in biological sciences in this one-day national conference. My discussion topic is in vitro testing of important samples. Why, which, and how. You can see the Department of Zoology and Gujarat University image in this very first slide where I'm going. I'm happy to share some more information and milestones achieved by my university with this highly intellectual audience, and then we will continue the today's topic. One more thing is that I am now hiding the visual of mine on the slide, so not have any errors during visualization of the content of upcoming slide. The oldest university of the Gujarat state was established in 1949 by the vision of great leader and freedom fighter King Sardar Vallabhbhai Patel. Gujarat University expanded in 270 acres of campus and completed 73 years of excellent academic journey. It has 23 schools, 65 departments, and 4 incubator centers, 6 centers of excellence along the way. It also includes the top 100 institutes of India by the NIRF ranking in 2021, and also included the top 10 government universities of India by ARIIA, that is Atal Ranking of Institutions on Innovation Achievements. One more feather in the cap of Gujarat University is that Gujarat State Institutional Ranking Paper, that is GSIRF, rated this university as a five-star university and given first rank among all the universities of the Gujarat. With a small introduction regarding my Gujarat University, let's start today's talk on in vitro testing of important samples. Why, which, and how. Outline of today's discussion, and it is an overview. Cell versus non-cell based assay. Give the answer about why in vitro testing of important samples are required. Then we will move on to which cell line and parameters we have to select to complete our research objectives associated to in vitro testing for our sample, and at last we will see cytotoxicity, genotoxicity, and functional assays along with advances in vitro testing systems. Understand about how we can plan or work related to vitro testing of important systems. Okay, so to begin with, why the in vitro testing? Let's first understand little bit about the convergence of various disciplines when there is drug discovery. It includes the various aspects like safety, design, novel molecule, or another pharmacokinetics, in vivo testing, and so on. These aspects intertwine with various disciplines like biochemistry, pharmacology, immunology, enzymology, physical chemistry, synthetic chemistry, information technology, the pharmacology, pathology, and behavioral science of in vivo activity, along with long dealing with intellectual property. But to start with molecule or our important sample, we must consider few aspects in of drug development, and they are time structural activity, pharmacological research, along with safety of the sample, and to deal with it, we must go through bioinformatics and in vitro testing. Directly jump to check in vivo activity will lead to consumption of lots of money, and it is also time consuming to handle the analyze the big data. Model to study the test compounds. The answers are very little number of studies can be done on humans. It allows for controlled activity, environmental variables, dosage or route of exposure can be controlled, experiments can be replicated, etc. But when we plan or design the testing of any compound for its non-cancerous effects through in vivo approach, we require large number of animals, have low throughput, it will be expensive, time consuming, and those response extrapolation will sometimes mislead researchers in some other direction. Many times it will be little focus on mode of action and biology and how few epidemiology studies. To cut a long story short, money and time consuming. If we see the actual process of new promising compound or drug, it includes in vitro and in vivo preclinical and then clinical testing. This is functional or biological assessment in drug discovery whenever new pharmaceutical drug development process. It has various steps in which recently added dry lab in silico stimulation to study the interaction of various targeted biological molecules, drug molecules, and in vitro toxicity testing experiments pave the way towards production of less expensive and targeted drug molecules for human experimental. Let's see an example which proves the importance of in silico testing. The example is of thalidomide. Introduction of thalidomide to overcome morning sickness, uneasiness, and nausea in pregnant women during late 1950s and early 1960s that leads thousands of ophelia cases in which pain deformities of the limbs occur. It was happened due to without studying teratogenesis or fetal safety. The thalidomide enters the market testing protocol to produce this kind of products. So now we know that in silico and in vitro, both approaches are very important and necessary for any compound before it comes into the market. The single molecule based screening and cell culture based high content screening is appear almost 100,000 per day, and tissue or lower organism based screening not more than thousands. On the other hand, there were only studies carried out on mammals. So, throughput potential for data acquisition is less when we go from molecular screening toward actual clinical trial. But on the same time, the human relevance of the data is tremendously high, and sometimes novel compounds fail when they reach this point. So, proper screening of new compounds before going to clinical trials and in vitro experiments will help to save the time and money. In this talk, I am majorly covering the in vitro assay and that is cell-based assay. Actually, we have two endpoints linked to molecule development. Cell-based assays are the bridge between these two, that is chemical tests and biological tests, or we can say a clinical trial. Cell-based bioassay represent a space between drug composition and the clinical trial. That means it is nothing but the connection between molecule development in chemistry and clinical trial in biology. So, what are cell-based assays? A bioassay is defined as an analytical procedure which analyzing and measuring a biological activity of a test substances like molecules or biopolymer or nanomaterial or even the test based on a specific functional and biological response of a test compound. They include testing the effects of molecules related to macrosis, apoptosis, cell viability, proliferation, signaling, transpiration, migration, ROS production. These are the list of few mostly performed bioassay among long list of cell-based bioassays. Major question immediately comes into our mind: why do we require them? Biologies are complex and heterogeneous in composition and can exist in several physical and chemical conformations that can impact on product safety and efficacy. By measuring the potency, we can infer the structural integrity of complex biologics. Thus, bioassays are measuring of quality of the therapeutic. And hence, we must perform this bioassay for clinical translation of new compounds by fulfilling the formulation attributes and biological attributes. It is not like that the bioassay or in vitro cell-based assays are having the answer of each and every question. A bioassay can, number one, indicate product quality. Number two, be relevant to mechanism of action and give insight into the way a therapeutic functions at a molecular level. And number three, precise and suitable for use in a environment. On the other hand, a bioassay cannot directly predict clinical efficacy or outcome. As for example, biodistribution, pharmacokinetic, non-specific interactions, anti-drug antibodies, etc. If we compare the pros and cons of cell-based versus non-cell-based assay, we can see that the cell line-based assay are closer to physiological conditions and followed by the scientists worldwide. More assays formats are also available to choose for the cell line-based bioassay, whereas non-cell line-based assays have less variability, easier to develop, and robust experimental design. These are the pros of cell line based assay and non cell line based assay. On the other hand, if we talk about the disadvantages, then higher variability and complexity. So, experience required to design experiment for cell line based assays, whereas the lesser functional characterization for non-cell line based assay is limitation of non-cell line based assay. Limitations are due to the complexity of cells and its nature to learn how to modify themselves. If we consider the cells in bioassay as one reagent among all other reagents, then it is the most critical and difficult to control for bioassay performance, and it has large contributors of bioassay variables. Hence, so critical but difficult to answer the questions are: number one, why can cells be difficult to control? And second, what defines a healthy, well-behaved cell? So cells are complex systems. Just to give you a glimpse of complexity, here we are going to see some of the numerical data related to human cells. Each cell have approximately 20,000 per haploid genome, which contributes approximately 1.5% of genome. But see the power of synthesis of not less than 40 million molecules per second. These different proteins perform specific tasks and have different locations based on the task. So cell-based bioassays are having its own limitations, but still they are highly sensitive because as compared to clinical trials in which the molecule tested on 50 multiplied by 10 to 12 cells, cell-based assay have approximately 25 multiplied by 10 to cells per well, which is million times less than cell present in humans. So, multiple assay systems can be performed using less number of cells, and we have data which we can average and provides rapid and effective means of screening and ranking chemicals. Is also essential for bridging between experimental animals and humans and for detailed understanding of the basis of species differences. It provides well-defined system for studying structure and in this slide, I also list out the limitations of in vitro model systems. They are actually not the real thing. Typically, static or non-active model system, systematic laws of phenotypic properties or functions of original tissues and cells are there. Similar liabilities with immortalized cell line is also there. We use similar kind of materials, instrumentations, and all other means which we require for immortalized cell lines and in vitro model systems. In vivo relevance often decreases with the simplicity of the model system. So, these are the limitations of in vitro model systems. Okay, this is just to introduce what is cell culture. The tissue or outgrowth from the primary explant is disposed clinically or enzymatically into a cell suspension, which may then be cultured as an adherent monolayer on a solid substrate or as a suspension in the culture medium. It has its own advantages and disadvantages. These are very basic information, but very important for graduate or postgraduate. I will go little bit quick for upcoming slides since this information is included in many of the graduate and postgraduate. We generally classify cultures, primary and continuous culture. Primary cultures derive directly from exercise tissue and culture. It has its own advantages and disadvantages. One of the major disadvantages is it can be maintained in vitro conditions only for a limited period of time. The continuous cultures derive from subculture of primary culture, which is also known as passage. There are two types of continuous culture, that is innate cell line and continuous or immortal cell line. Innate cell line have innate life, senescence after approximately 30 cycles of division, usually deploy and maintain some degree of division. On the other hand, continuous cell line can be propagated indefinitely, but it retains very little of the original character. So, we cannot have a good test model if we have continuous cell line for exactly matching the original cell. These are the list of some commonly used cell lines derived from human and primates. As per our requirement of the target tissue for our newly synthesized or extracted molecule, we can select particular cell line for test. If we want to carry out research on pesticide and its toxicity, then also we have a specific cell line available from insect cells. They are SF9 and SF21. are the images of oldest and commonly used cell line derived from cervical cancer cell and named after Henrietta Lacks, the 31-year-old African American lady who died due to cancer. These cells can be procured from that is American Type Culture Collection or NCCS, that is National Center for Cell Sciences. This images also shows the low density on the left side of the image and good congruence on the right side of the image of the HeLa cells. Research students should keep track and harvest the cells and extremely 25% of congruence for various in vitro experiments. We should also know where do we begin if the compound or molecule in our primary search shows characteristic to bind on the plasma membrane, then go happy and present on membrane which involves cascade or various participating the caspase family protein and ultimately responsible for the apoptosis. If in the primary search the compound identified as crossing the plasma membrane barrier and act in the cytoplasm, then we must study the intrinsic pathways. The intrinsic pathways involve mainly the effects on mitochondria along with the alteration in the function of lysosome, ER complex, and other cell organs. And on the nucleus and genetic material, mostly it crosses the nuclear membrane. So, what kind of compound or what kind of characteristic of the compound we have, that is very important to decide which kind of pathway and how many pathways to kind of study. So, to decide the whole program of the study and formulate the plan of work, we must consider the questions like what is the objective of experiment, what is the hypothesis of experiment, what is that we are looking for, whether it is on the cell membrane or it is in the cytoplasm or it is in the nucleus and attached to the DNA. Where is that we are looking for? That is also what. So, to understand where do we begin, let's think in some different directions. It is not only the targeting the pathways, but it is also equally important to choose the relevant bioassay to study those pathways, since there are many relevant bioassays available to prove the similar endpoints. As for example, the growth of cell inhibited by altering the growth factors activity or by the cytokines effect, especially on immune and inflammation responses. These cytokines also help to boost anti-cancer activity by sending signals that can help make abnormal cells die and normal cells live longer. So, the effects are on both extreme endpoints, and researchers should decide the critical biological molecule and also think in direction that whether that biological molecule can be exploited to develop the bioassay. It is not only to design the relevant bioassay, but also simultaneously require the right reagents for the right bioassay is very important. Sometimes little compromise on purity of the reagents will totally shift the final result and mislead the experimenter to some other direction, which lead to either death end of the research or circular or linear non-concluded study results. I mean to say, researchers either come again on the same questions where the experiments start, that is circular, or the end point is not visible and endless experimental cascade begin to be ruled out. The null hypothesis, that is linear non-concluded study. Moreover, along with the finalization of intrinsic and extrinsic pathways to be studied, is equally important to choose the relevant and reliable cell type for study. So, these are the important questions to start the journey. These questions should be asked to the individuals who want to test the molecule. It will help to the researchers to design the assay. If you have chemical synthesized compound or herbal extracts or formulations or nanoparticles, then check the properties of it like solubility, size, probable reaction site in the cell, and probable biomolecule to which it attached. If molecule is water soluble, then those can be given in culture media, and if molecule is hydrophobic, then DMSO can be used to all the molecules. If we use DMSO, then we have the limitation to increase the concentration of the test molecule because we cannot exceed the 0.5% of DMSO concentration in final test volume. That is maximum 1 microliter in 10 microliter of dose in total 200 microliter of test volume. Otherwise, it will give its own toxicity. So, frequent, vital, and dynamic communication between both researchers. Number one, molecule synthesizer, and number two, test performer must be required. This is the flow of cell-based assay. Generally, we first check the cytotoxicity of the nucleoside molecule or chemical with various cytotoxicity testing parameters, including MTP, XTP, SRPSA, and so on. If we assuming that the test molecule having the property to damage the nucleic acid indirectly or directly, then we can perform genotoxicity testing. In genotoxicity, in vitro testing, we have various parameters like chromosomal aberration, ferment assay, micronucleus and DNA ladder formation, along with effects on UC19 factors or helix DNA. Result of these assays give us information that whether the test molecule is toxic or not. The required functional assays as per target tissues can also be performed at later stage using in vitro cell-based assays. Let's check some important information related to these assays, that is cytotoxicity assays, genotoxicity assays, and functional assays in few upcoming slides. This slide is indicating the target time for cytotoxicity testing. As for example, to check the effect on lysosomal activity, we can perform acid phosphatase and neutral rate assay. If we are assuming on the basis of literature review that our test compound have effect on membrane permeability, then we have neutral rate and LDH assay to analyze it. To evaluate XRV assay or crystal violet assay. To assess mitochondrial effect, we can implement MTT or XTP assay. In short, we have different in vitro cell-based cytotoxicity assay system to estimate the effect of test sample on target biocomponent in live cells or dead cells. Okay. This slide indicates various genotoxicity test systems depending on the hypothesis related to effect of test compound on genetic material. If we hypothesize that our test compound can be responsible for mutation or double strand breaks or having interaction with the mitotic apparatus, then we have different genotoxicity testing parameters like Ames test or micronucleus acid or chromosomal aberrations test respectively. Check the direct interaction and damaging properties of test compound. We have tests on TUC19 method or we can perform Eric's sperm DNA damage test. Regarding the functional assays, we have batteries of assays and among them, one example is shown in this slide. There are kits available to check whether the test compound having the effect of reduction in cell viability, apoptosis, or necrosis using Usha's probe or using cytometer. Like if high LDH is detected in cell culture medium as compared to control group, then cell having necrotic effect. And if cell having a flapping, low AKT and alteration in phosphate 3, then it is entered into the program cell death, also known as apoptosis. This is only the glimpse of functional assay. Actually, we have functional assay targeted to check the effects on various organelles and biomolecules as well. And it is not possible to explain many assays within a short period of time. Okay, so the last objective of this presentation is that we will see the advanced in vitro testing system, understand about how we can plan of our work related to in vitro testing of important samples. Most of these advanced in vitro testing protocols or kit-based multi-endpoint SOPs are applying the fluorescence-based detection system using either fluorescent microscope and dedicated software or using highly precise broad-range wavelength high throughput multi-mode reader or using fluorescent stack probe or antibody detection system in cytometer. As I already described previously, we have different cytotoxicity measurement endpoints, and it can be possible to use it to check cancer cell viability or proliferation. Standard SOPs available in kit-based protocols can also detect the oxidative stress or effect of reactive oxygen species of various markers like A-isoprene, A-methylation, protein carbonylation, glutathione, etc. And also measure detection of enzymes activity like catalase, superoxide dismutase, glutathione S transferase, etc. Numerous companies manufacture kits which either UV or visible light detection system. Different high throughput detection systems mostly work on the three stages of cell signaling process. It checks whether the test compound have any activity at cell membrane reception site using identification of binding of test compound with specific receptor. It also checks whether test compound, if enter the cell, then what kind of signal transduction pathway it follow, and lastly it also checks the activation of cellular processes directly or indirectly by checking mRNA synthesis or presence of specific. Let's see two examples of this kind of detection system. There is an acute need for the development of a rapid technique that can use the physical and early clinical phases of drug development. Collins Biosciences Company, registered at San Francisco in California, was developer of a three-dimensional cell culture microarray technology intended to provide the high throughput screening of both compounds and their metabolites for toxicity. The company technologies use a biocatalytic platform to profile chemical compound toxicity to enhance decision making or supporting new product development in the pharmaceutical, biotechnology, cosmetic, and agri-chemical industries, enabling researchers to improve the process of new compound development. This is the another example. This is cell culture based quantitative high throughput. Very first left upper image shows that three robotic arms on the robotic system that enables ultra high throughput of small molecules against diverse type of gases. The automated unattended screening arms strips the 1,500 surface well plate format microtiter plate and puts them among the various sections of the problem. The practical implementation of this quantitative high throughput screening for cell-based and biochemical assays across libraries of more than 1 lakh compounds required maximal efficiency with minimization of the assay system and the ability to easily accommodate many different assays format and screening protocols. Okay, so let's see engineered tissue-based. The left side schematic illustration of the microfluidic-based cell culture platform incorporating the patterned cells with a micro-injection array for drug testing. The other two images are a fused multi-well plate in an array of bioreactor integrated in a multi-well plate format. It fosters long-term maintenance of fluidity under constant microperfusions, mimicking in several important ways the complexity and 3D microenvironment of people. It is assumed that the better a cell culture system mimics the complexity and microenvironment of a tissue or an organ, the more useful and broader information it can provide. These are the list of commercially available cell-based engineered and newer model for drug development. It comes with frozen or model and just heating it at desired temperature. Put the acid plate along with acid solution provided by the company and give the data to software after stipulated time interval. Software analyze the data and give multi points, multi-parametric output with interpretation. side is the overview of current and principles for single cell selection, which includes traditional tools like fluorescence activated cell sorting, laser capture, microdissection, and micromanipulation, and modern tools like microwell, microfluidics, capillaries, etc. The illustration on right side is a high throughput targeted transcriptomics approach in Nanovell's cartridge-based Rhapsody platform of Giri Bioscience generate protein and transcript activation data on the single cell level for more than 10 more single cells, gentle but robust microwell-based single-cell partitioning technology. At Department of Zoology, Gujarat are fortunate enough to have this 3D Rhapsody platform in our department. So I am concluding my talk with the remark that in vitro analysis of the effect of test compounds on all biochemical and genomic variables in culture of interest tissue to the single cell for development of a new drug molecule and its toxicity testing is not a far and it will be routine approach in pharmaceutical and basic academic research. I am thankful to ALE Society and the chairman of this society, Dr. Prabhakar More, session chairperson, Dr. S. Katlam, organizing committee chairperson and principal, Dr. G. Convener and head of department of zoology, Dr. Usha Karunakaran, organizing secretary and assistant professor, Dr. Shashikala Prajapati, along with all team members, Department of Zoology and IQAC, and last but not the least, participants of this one-day national conference on frontiers in biological science. Have happy learning and thank you for this.
Thank you, sir, for your encouraging words and setting the perfect background for the conference. Before we start with the technical session, I here declare the end of the inaugural session. We will begin with the technical session after 30 minutes on the same link.
Be ready with your presentation.
Welcome back to one day national e-conference on frontiers in biological sciences. Participants of the conference are requested to unmute themselves if they have any questions. Ahead with our today's presentation session, we have with us Dr. Savitri Katlam, Assistant Professor, Department of Pharmacology, MGM Hospital, Kamothi, Navi Mumbai, who will review the technical session. I welcome you, ma'am, to our national conference. I request all the participants to be ready with their presentation. Very first, I request Preeti Singh from Ram Niranjan Jhunjhunwala College of Arts and Science, Commerce College from Ghatkopar, to present her research paper on bioaccumulation of microplastic fibers in the gut of Sardinella longiceps, collected from Sashunda, west coast of Maharashtra. Over to you, ma'am.
Very good afternoon, ma'am.
Preeti ma'am, you are not audible. Hello.
Yeah, ma'am. Am I audible and visible? Actually, I'm presenting my screen in my phone, so I'm not that much confident, ma'am. If any issue is there, please let me know.
Yeah,
Hello.
sure. Yeah, you
Yeah.
can start now.
Okay. Yeah. Thank you.
Ma'am, voice is not coming. Just see now. Preeti ma'am, you're
Yes,
not
ma'am.
audible. You're not audible.
I'm not audible. Oh, okay.
Yeah.
Ma'am, if possible, can you only play the video I have given, please, ma'am? Hello. I'm sorry, ma'am. I think the voice is not clear.
Yes, ma'am, I will share your video. Just wait.
Thank you so much, ma'am.
आवाज नहीं स्टॉप करो आवाज नहीं आ रही है चेक करो
मुझे आ रही है.
आपको आ रही है आवाज
हाँ. करूँ कंटिन्यू?
यूट्यूब पे चेक करो आवाज आ रही है मुझे आवाज नहीं आ रही यूट्यूब पे आ रही है आवाज एक सेकंड स्टार्ट करने के लिए तुम आवाज चेक करो यूट्यूब पे
जब आता है
नहीं आई तो
गूगल मीट पे नहीं आ रहा है ठीक क्या आवाज नहीं आ रही
मैंने स्क्रीन शेयर किया. मुझे आवाज आ रही है.
आपका स्पीकर ऑन है चेक करो
प्रॉब्लम नहीं
वॉल्यूम चेक करो
फुल है. मेरा हेडफोन लगा इसकी वजह से.
ऑन है क्या स्पीकर देखो ये
अरे उनको माइक क्यों दे रहे हो उनको माइक चालू करना
माइक ऑन करो
माइक ऑन है. ये देखो ना, ऑन है.
मैं इधर ऑन दिखा
आप दिखा रहे हैं किसी का माइक उनका माइक ऑन करना पड़ेगा बोलो
कैमरा ऑफ किया. देखना माइक ऑन है भाई. से माइक इज ऑन.
नाम क्या है
प्रीति.
बंद
अरे मैं उनका वीडियो प्ले कर रही हूँ ना. ये है
ये
वीडियो.
सुनाई दे रहा है मुझे
ओके.
प्ले करना ओके When technical issues are there, we are solving within 1 or 2 minutes.
There are some technical issues we are solving in just 1 and 2 minutes. वीडियो रहने दो मेरा।
वीडियो अभी नहीं, हम जाने के बाद
मोबाइल पे करो, मोबाइल पे
ओके।
Very good morning to one and all present here. This is Preeti Singh, a PhD research scholar from Ramniranjan Bhimwala College, Mumbai University. Today I am going to present bioaccumulation of
ये क्यों हो रहा है?
microplastic fibers in the gut of Sardinella longiceps. Before starting my presentation, I would like to thank KLE College Zoology Department for giving this platform to present our work. Let us start. Bioaccumulation of microplastic fibers in the gut of Sardinella longiceps, collected from Sassoon, west coast of Maharashtra. As due to increasing human population, anthropogenic activities are also increasing, leading to littering of plastic in buried biota. This plastic degrades into microplastic. As most of the microplastics are colorful, they mimic prey or marine organisms and get bioaccumulated in different organs of the fish. When we humans eat these seafood, microplastics get ingested inside the human. By this circulatory process, it gets into the food web. This is the sampling side. Let us start to material method. The sampling side we have used is Sassoon. Percent KOH. Filtration is done to filter the filtrate and oven dry at 60 degrees centigrade for 48 hours. Moving ahead, screening is done on stereo microscope where the morphology and the structure, shape, color of the microplastic found is seen. The analysis is done on ATR-FTI spectroscopy where the particles are analyzed and the name of the particles are seen. This is the setup of our college. where we have done the analysis. Here you can see the pseudo microscope and ATR machine, and in the computer, the library, spectral library is there, which shows us the presence of microplastic and the type of microplastic. Precaution we have to take that cotton, pure cotton, apron and gloves, all these precautionary measures to be taken to minimize the false positive result. Moving ahead, as a result, we have found different colors of microplastics of different shape and size and color. We can see pink color, yellow, blue, brown and black color. All these are found in the gut of Sardinella longicea, which is collected from suckling dogs. Moving ahead, this is the FTIR report where we have found that polyamide resin is most commonly found in the gut of sample as a microplastic. The different frequencies wavelength present, polyamide resin is present in the sample. Moving ahead. The shape we have found is slender, comma-shaped and curved shape of microplastic. The size varies from 0.1 mm to 2.5 mm. The microplastic other than polyamide we have found is polyethylene, polypropylene, polystyrene and polycaprolactam. Moving ahead to the conclusion of our work, we found that there is bioaccumulation of microplastic fiber in the gut of Sardinella longiceps, which is collected from Sassoon Dog, Mumbai. The size we have found is 0.1 mm to 2.5 mm. The color which is more frequently found were blue and red color. Blue color may be because of the seed. Polyamide found more pronounced. The stereo microscope along with ATR FTIR is a useful tool we found in detection of microplastics. We need to understand the plastic waste management for betterment of future of mankind. I would like to acknowledge Dr. Leena Muralidharan, my guide, the principal of Ramiranjan Jununwala College, Dr. Himanshu Danga, and non-teaching staff of Ramiranjan Jununwala College for helping me throughout my experimental work. These are the references we have used during our study. Considering the harmful impacts of microplastics, it is recommended to develop proper solid waste management in marine ecosystem to prevent ourselves getting in plastic cage. Swachh Bharat, Swasth Bharat. Thank you.
Thank you, ma'am, for your presentation. Thank you so much.
Thank you so much, ma'am, for helping me.
Moving ahead, our next presentation is by Bhavna Mahesh Bhai from Gujarat University and Department of Zoology. Her topic is checklist of plankton of two different freshwater bodies around Dahod, district Gujarat, India. Participants are requested to be ready with their presentation. Over to you, ma'am. Yes, Bhavna Mahesh Bhai, Pargi. Are you there, ma'am? Next presentation will be by Priyanka Waghela from Department of Zoology, School of Sciences, Gujarat University. Her topic is Study of Plankton Diversity in Dholka Village, Ponds of Ahmedabad, District of Gujarat. Priyanka, ma'am.
Dear participants, kindly be ready with your presentation. In fact, we have given you the sequence also as to who will be presenting first, second, third, and so on. So kindly be ready with your presentation. Thank you.
Yes, dear participants, be ready with your presentation. Priyanka Waghela and Bhavna Mahesh Bhai, are you there? Moving ahead, we will start with next presentation by Rajeshri Rajesh Sawant from Maharshi Dayanand College of Arts, Science and Commerce, Parel. Her topic is characterization and antimicrobial activity of silver nanoparticles synthesized using scales of Indian salmon. Over to you, ma'am.
Yeah, good afternoon, ma'am. Am I audible to everyone?
Yes, ma'am, you are audible.
Yeah, thank you very much. Actually, I have joined from two devices, basically because my camera is not working off my laptop.
Okay, ma'am.
Okay, yeah, I'm presenting now.
Yes, ma'am, sure.
Ma'am, presentation is visible. Ma'am, my screen is visible.
Yeah, you are speaking, that screen is visible.
Yes, ma'am, that is the one.
Yeah.
Okay. Good afternoon, everyone. This is Rajeshri Sawant from MD College. Topic of my presentation is characterization and antimicrobial activity of silver nanoparticles synthesized using scales of Indian salmon. Its scientific name is Eleutheranima tetradactylum.
Rajeshri, ma'am, your
Yes.
PPT is not visible to us. Your PPT is not visible to us.
Okay, ma'am, I will log out from this device and I'll just keep one device only, the one
Yeah,
which
sure.
have the PPT. Okay, ma'am.
Yeah, sure.
Yeah, thank you. Ma'am, is this visible?
No, ma'am, not.
1 minute, ma'am.
Ma'am, you present your screen now.
Yeah. Hello, ma'am. Hello, ma'am. I'm audible?
Yeah, you are audible.
Okay, ma'am. I'm presenting it again.
Okay.
Ma'am, is this visible?
No, ma'am, we can't see your screen, ma'am.
1 minute.
Ma'am, share your entire screen. Yeah, now it is visible. Yeah.
Yes.
Yes. Yeah.
Thank you very much.
Yeah. Start with slideshow, ma'am. Huh?
Yes, ma'am.
Yeah.
Yeah. This is the title of my paper, Characterization and Antimicrobial Activity of Silver Nanoparticles.
Ma'am, slideshow, please. Ma'am, slideshow. Start करिए ना, please.
Yeah.
Yeah.
So, introduction. Recently, researchers adopted the green synthesis of metal nanoparticles to overcome the challenges posed by the hazardous chemical synthesis and the energy expensive physical methods. Green synthesis of metal nanoparticles include use of microorganisms such as bacteria, fungi, viruses, and plant extracts. Extensive research is performed on green synthesis of metallic nanoparticles. However, literature on the use of animal extracts is rare. Our study focuses on the green synthesis of silver nanoparticles using aqueous extracts of scale of Indian salmon fish, that is Eleutherinema tetradactylum. India generates more than 2 metric million tons of fish waste annually, which includes skin, viscera, fish scales, and bones. Thus, it has become an increasing environmental concern due to its high disposal cost. Hence, it is necessary to acquire sustainable, eco-friendly, and less expensive recycling methods which can assure reuse of fish waste. Fish scales account for around 6 to 7% of fish waste and is known to contain bioactive components such as proteins, which includes collagen and gelatin, and minerals such as hydroxyapatite. Here in this slide, you can see the percentage of waste generated, and other side there is a picture of the fish waste. The next slide, which is there, the next slide shows the.
Ma'am.
Yes, ma'am.
Ma'am, your slide is not moving. It is just on the first slide only.
Is it so?
Yeah.
Ma'am, 1 minute. Huh? Ma'am, I will do it manually. Is it okay?
Yeah, ma'am. Just start with your this slide show, na? So bigger screen will be there, ma'am.
Yeah, I already started with that. Okay. Now this is visible.
Yeah.
This is the first page, ma'am. Is it visible?
Yeah, fourth plate we can see. Fourth one.
Okay. I have started it again, and this is the third page. Now I'm on the fourth page, ma'am.
Continue, ma'am. Continue.
Yeah, I'm extremely sorry.
Ma'am, click F5.
Yeah, this is the fifth slide. which shows the components of waste, which includes head, then skin, scales, bone, that is in the percentage. My next slide, which is there, using extracts from fish waste, such as scales, which constitute 6 to 7% of total fish waste, can be an alternative technique for chemical synthesis, green synthesis of metal nanoparticles. Hence, present studies propose to develop an eco-friendly, clean, non-toxic, facile, chemically preparative method for metal nanoparticles using the extract of fish scales, acting as reducing as well as the stabilizing agent, and to study the antimicrobial activity of novel silver nanoparticles against various pathogenic organisms for potential application in medical and allied industry. Ma'am, this is visible. Ma'am, the slide is visible.
Yes, madam, we can see.
Okay, yeah, thank you very much. Now the next slide, which is there here, you can see the picture of Rawas fish, that is Indian salmon fish. I have removed fish scales from it, and this fish scales we have cleaned, washed with the distilled water, and then we have prepared the extract from the fish scales. In the next slide, you can see that here the fish scale extract and the silver nitrate solution is mixed, which results in the formation of silver nanoparticles. And here you can see in the resultant solutions, there is increasing color intensity of the silver nanoparticles, where we have taken different concentrations of fish scale extract. After 24 hours, when the photographs were taken, we can see that the test tubes, where the first test tube is of the control, there is no reaction, but otherwise the other 4 test tubes, which include 10% to 40% different concentrations of FSE, it shows the color change, that is the brown coloration. Results and discussion. Treatment of silver nitrate with fish scale extract for 3 days showed development of brown color with the mixtures as a clear suspension indicating formation of silver nanoparticles. The color intensity in reaction mixture gradually increased with increasing concentration of fish scale extract, that is from 10% to 30%. The UV visible spectra, where the wavelength was ranging between 300 to 800 nanometer when taken, it shows the absorbance peak at 430 to 440 nanometer for different concentrations of fish scale extract. Here we can see the graph of UV visible spectroscopy, where it shows that 10% fish scale extract use that shows the peak between 430 to 440 nanometer. For the second sample, where the fish scale extract concentration was 20%, there it shows the peak of 435 nanometer, whereas the third one, which is there, that shows, that is for the 30% of FSC, it shows the peak between 430 to 435 nanometer, whereas the overall UV range for the silver nanoparticles is 400 to 500 nanometer. We have taken the transmission electron microscopy images. Here in the next slide, you can see that the nanoparticles which have been synthesized, they are from 10 nanometer to the 60 nanometer. The second diagram which is there, it shows the selected area electron diffraction analysis of silver nanoparticles, which was prepared using 30% of fish scale extract. Now about the applicational part, antibacterial activity. Here you can see that the strains which were selected, they were Escherichia coli, Salmonella typhi, and Staphylococcus aureus. Out of them, Escherichia coli and Salmonella typhi, they were susceptible to the given nanoparticles, whereas Staphylococcus aureus has been seen resistant to the same. Conclusion of the presentation. Present study confirms that bioactive molecules such as collagen and gelatin present in extract of scales of Indian salmon have potential to reduce and stabilize the silver nanoparticles. The formation of silver nanoparticles was confirmed using UV visible spectroscopy with maximum absorbance in the range of 430 to 470 nanometer, and the TEM images revealed the formation of polydispersed silver nanoparticles which were of various sizes such as spherical, triangular, and cubical, and they were ranging between 6 to 70 nanometer. The silver nanoparticles also demonstrated antimicrobial activity against E. coli and S. typhi, indicating its potential for application in biomedical and healthcare industry. The present study demonstrates an alternative for fish waste management by using aqueous extracts of fish scales for eco-friendly, clean, non-toxic, and efficient silver nanoparticle synthesis. Next slide is of the reference literature. And here it ends with my presentation. Thank you very much.
Thank you, ma'am. Thank you so much. The next presentation will be by Bhavana Mahesh Bhai Pargi from Gujarat University and Department of Zoology. Checklist of planktons of two different freshwater bodies around Dahod district, Gujarat, India. Over to you, ma'am. Okay, we'll go ahead with next presentation. Bhavna ma'am, are you ready? Hello? Bhavna Pargi ma'am, are you ready with your presentation? You're not audible, ma'am. Anyways, meanwhile, we'll go ahead. You keep yourself ready with your presentation. Next presentation will be by Saili Roshan Kamble from ICL Motilal Jinjunwala College, Vashi. Her topic is to study seasonal variation in physicochemical parameters of Karve Lake, Nerul, Navi Mumbai, India. Over to you, ma'am. Ma'am, you are not audible. Please unmute yourself.
Am I audible now?
Yeah, you are audible.
A warm good afternoon to all the respected dignitaries and all of you present here. Myself, Saili Kamble. I am from ICL Motilal Junjunwala College. I will start with my presentation. Is my screen visible to everyone? Am I audible?
Yes, ma'am. Yes,
Is
ma'am.
my screen visible?
Yes.
My topic of the presentation is to study the seasonal variation in physicochemical parameters of Karave Lake, Nerul, Navi Mumbai. So this is the picture of the lake which I'm working on. So basically, you all know all biological lives and their nourishment is dependent on the water. Sustainable development will be impossible without fresh water. Lakes are one of the most valuable resources, ecosystems, because they support and maintain a balanced adaptive community of organisms with a huge species richness and unique biotic integrity due to functional organization of all organisms. Lakes, a key aquatic environment with life supporting system, have been experiencing ecological degradation for past 2 decades as a result of physical, chemical, biological and unwanted anthropogenic activity. Navi Mumbai is one of the fastest growing city in the world and it is blessed with numerous lakes that serve as a major natural resource because it is densely populated and it has a significant impact on the health of the water body. So aim of my work was to determine the seasonal fluctuation in physicochemical characteristics of Karave Lake. The objective of my work was to study the seasonal variations in physicochemical characteristics in the water body of Karave Lake in the Navi Mumbai city. This you can see the topography of Karave Lake, wherein it shows 19.14 latitude, whereas 73 longitude. Material and methods. During the study, water samples for the analysis was collected for the period of 1 year from the month August 2018 to July 2019. Water sample was collected from 5 different sites in the well-rinsed plastic cans between the time 9 to 11 a.m. This you can see 5 slides from where I've collected the water samples. Water samples around 1000 ml was collected. Data was compiled for post-monsoon, pre-monsoon, during monsoon and post-monsoon. 14 important physicochemical parameters were analyzed for water quality. Analysis were performed following AFA 2012. So these are methods which I have applied. For temperature, thermometric method. For pH, digital pH meter was used. In situ, dissolved oxygen, Winkler's method. BOD, Winkler's method. COD, sodium thiosulfate method. Free carbon dioxide, titrimetric method. Acidity, alkalinity, again titrimetric method. Then for nutrients, phosphate, phosphorus, colorimetric method. Then nitrate and nitrite, NADD method was used. So this is my compiled data where it shows the reading for pre-monsoon, monsoon and post-monsoon. Let's move to the histogram to see the data analysis. Here first we can see BOD. BOD during monsoon season, it showed the higher value. BOD can be utilized as an indicator of organic matter. So BOD was done. BOD less than 1 milligram per liter. It reflects that water is unpolluted. Whereas moderate BOD ranging from 2 to 9 milligrams per liter, it shows that water is moderately polluted. More than 10, it shows the water is highly polluted. Then electrical conductivity. Electrical conductivity of water increases due to concentration of salts. It is also a very good indicator of water pollution. So it shows that during monsoon, the level of electrical conductivity was high as compared to pre-monsoon and post-monsoon. The next parameter which was undertaken was salinity. The salinity is one of the most important and essential factor for the water body. It also shows the presence of aquatic life. It is important factor for the aquatic life. The high salinity reflects the presence of inorganic dissolved salts. The results showed that post monsoon, the salinity was high as compared to pre monsoon and during monsoon. When it comes to pH, pH is one of the most important critical physical parameter. So during my study, it was found that the pH was more or less found to be same, ranging from 7 to 8. DO, am I audible?
Yes, ma'am.
Thank you so much. DO, dissolved oxygen. It is one of the most important parameter because dissolved oxygen shows or reflects the good health of the lake. So, and the DO is less than 2 milligrams per liter. It reflects that the extinction of organisms. So, during my study, the DO was found to be kind of equal in all three seasons. During monsoon, it was seen that there was elevation in the DO level, dissolved oxygen level. TDS, total dissolved solids, was again found in all the three seasons. In all the three three levels, it was found that the TDS was found to be more or less same. It was little high during pre-monsoon, maybe due to evaporation ratio. Hardness during monsoon, it was found to be high as compared to pre-monsoon and post-monsoon. Am I audible?
Yes, ma'am, you are audible.
Thank you. Hardness during pre-monsoon and post-monsoon, it was found to be high. During monsoon, it was found to be high as compared to pre-monsoon and post-monsoon. Hardness is basically due to presence of cations, anions, carbonates, and bicarbonates. Free carbon dioxide. Free carbon dioxide is a result of respiration and decomposition of aquatic life. So, free carbon dioxide during the study, it was found to be kind at the similar level in all three seasons. Alkalinity. Alkalinity is the ability to neutralize the acid. So during the study, it was found that alkalinity post monsoon was high as compared to pre monsoon and during monsoon. Phosphate level, it is one of the most important parameter as it reflects the as it reflects the deposition of dead organisms within the lake. The phosphate level post monsoon was found to be high as compared to pre monsoon and during monsoon. Nitrate level. Nitrate is one of the biological it is one of the byproduct of biological oxidation of nitrogenous products. The level of nitrate was found to be equivalent, more or less equivalent during all three seasons. COD, chemical oxygen demand. Pre-monsoon, it was found more as compared to during monsoon and post-monsoon. Turbidity. Turbidity is a result creates turbidity during monsoon was found to be higher as compared to pre-monsoon and the post-monsoon season. The temperature was found to be less, maybe due to ambient temperature, as compared to pre-monsoon and post-monsoon. So with this, we wind up with the physicochemical parameters. Now the conclusion. Karave Lake is one of the tourist attraction of the Navi Mumbai. Lake, it is getting polluted day by day. An enormous variety of chemical compounds produced by human activities pollute natural bodies, causing both acute and chronic pollution. In this study, physicochemical variables showed considerable seasonal change. Parameters such as total dissolved solids, nitrates, chemical oxygen demand, temperature were shown high during pre-monsoon season, whereas biochemical oxygen demand, electrical conductivity, dissolved oxygen hardness, turbidity showed high value during monsoon period, and salinity pH, alkalinity, phosphate showed high value during post monsoon season. These are my references. I acknowledgement. I take this opportunity to thank respected principal, my guide, Dr. Anita Zadav, and entire Zoology Department of ICL Motilal Jhunjhunwala College of Vashi for helping me and guiding me through my work, and a big thanks to KLE College for giving me this opportunity to present my work here. Thank you so much.
Thank you, ma'am. Bhavna ma'am, are you ready?
Yes, I am audible.
Yeah, you are audible now.
Okay.
Yeah. You can start.
My PPT is shown or not?
Yeah, it is visible, ma'am. You can start.
Okay.
Yeah.
Good afternoon, everyone. Myself, Bhavna Pargi, and I am here to represent my research paper on checking of plankton of two different freshwater bodies around Dahod district, Gujarat. Introduction. Pond is a freshwater ecosystem in which the various organisms depends on one another and its play a key role in maintaining regional biodiversity. The term plankton refers to organisms which have drifting habitat and include all the forms of both macro and microscopic life, which float free in water or if motile are unable to swim against water current. Plankton is divided into two mainly parts, phytoplankton and zooplankton. Phytoplankton are free floating unicellular filamentous and colonial autotrophic forms of aquatic habitat whose movement is more or less dependent on water current. Freshwater community are very much sensitive to environmental variable. Creatures living in aquatic environment lead their existence in a balance of nature. This balance has been changed during the last decade due to the factors such as overfishing, eutrophication, and lack of oxygen. As a consequence, some zooplankton species are disappearing or available only in small number. Zooplankton are major mode of energy transfer between phytoplankton and fish. These are divided into different groups such as Rotifera, Cladocera, copepods, ostracods, and protozoa including their larval forms. Material and method. The sample were collected in the morning and evening time during between 7 to 9 am and pm. Plankton net having mesh size 20 and 50 micrometer. Then sample were fixed in 4% formalin. This proceed by Lugol solution glutaraldehyde stain used for phytoplankton and rose bengal stain used for zooplankton. The sample was allowed to settle down for 24 hours. A drop of water was taken on glass side and observed under light microscope attached to camera and using MOTIC image plus software. The identification of plankton by using Advention and Tonapi book and many other literature. Result and discussion. In both the pond, I have found 36 zooplankton species belonging to group 24 species of Rotifera, 7 species of Copapoda, 4 species of Cladocera, and 1 species of Ostracoda. Individually in Thala Pond, 29 genera I found, 4 groups in descending order, higher number of 24 Rotifera species, followed by 3 Copapoda species, 1 Cladocera, and 1 Ostracoda species. In Pathampur Pond, I found 17 species found. In this order of species community, it different by Thala Pond, which was 7 Copapoda, 6 Rotifera, 4 Cladocera species found. These are the name of zooplankton species: phylum Rotifera, family Brachioidae. Branchionus genus is. Am I audible?
Yes, ma'am, you are audible.
Okay. Brachionus forficula, Brachionus calciforus, Brachionus diversicornis, and Keratera tropica, Keratera tecta, which found in both the pond, Pathampur and Thala pond. Brachionus caudatus, Brachionus rubens, Brachionus asagachinesis, Brachionus chondridentus. These are the species of zooplankton. Keratella cerulata, Ergonotholca foliaceae. In family Esplanchidae, I found in Thala pond Esplanche herrici, Esplanche brandtwelli, Esplanche prydonta. The family Trichoseridae, I found Trichosera edmondsoni, Trichosera cylindrica. In Synechidae species, I found Polyarthra vulgaris. Procosperidae family, I found Testudilena species. Trochosperidae family, I found Philiana longista and Phiriana opilinenis. In phylum Arthropoda, subphylum Crustacea, class Brachiopoda, order Cladocera, family Daphniidae. In Thalapond, I found Daphnia becna. In Pathampur pond, Daphnia magna, Daphnia lumpoltizi, Daphnia axiom, and Daphnia sarsi. These both are in family Cididae. In class Copapoda, Cleonida, Fabelli Depnoimedae, Thalapond I found Heliodiptomus vidus. In Pathampur pond, Diloptomus vidus and Cynodiptomus indicus. In order Cyclopodia and Cyclopedia fabelli, I found in the Thalapond Thermocyclops helenus and nucleus species. In Pathampur pond, Thermocyclops decipiens nucleus species, Thermocyclops helenus, Mesocyclops leucati, and Dicyclops thomasi. In class Ostracoda, order Podocopida, Fabelli Cacrididae. Thala pond species name is Eucrypsis basiconesa. These are the name I have already discussed in table. In the present study, among the rotifers, the genus Brachionus was found to be more in numbers in Thalapond. Similar to these results found in M. Dharasekarna et al. In 2016, rotifers are the microscopic, soft-body, aquatic, multicellular invertebrates, which are the indicator of health. Thus, in the present study, dominance of rotifers was reported, which similar with several researchers. Crustacean zooplankton Cladocera, Copapoda, and Ostracoda act as secondary consumer in the aquatic food chain. Cladocera, also known as water fleas, are transparent and larger than rotifers. They have high nutrition value and existing food for secondary consumer. These are found plenty in ponds and lakes than rivers. Due to their easy culturing, short regeneration time, and clonal reproduction, Cladocera are important model organism in ecological and evolutionary study. In Pathampur pond, I found high diversity of Copapoda. Copapoda are important member of zooplankton for their role in the trophic dynamics and in energy transfer in aquatic ecosystem, provide food for fishes and freshwater ponds, lakes, and play a major role in fish production. Ostracoda species lower from all the group in both the pond. The ostracod are bivalve structure. They occur in all kind of freshwater and marine environment too. In phytoplankton, I found in checklist 32 overall species in both the pond. which belongs to 11 chlorophyceae, 14 species to bacillophyceae, 5 species to cyanophyceae, and 2 species to euglenophyceae. In Thalapon, total 26 phytoplankton I found. The species diversity consists 13 bacillophyceae, which is followed by 9 chlorophyceae, 3 cyanophyceae, and 1 euglenophyceae. In Pathampur pond, 19 total species I found. In this pond, species community represented by 8 bacillophyceae, 6 chlorophyceae, 3 cyanophyceae, and 2 euglenophyceae species. Similar result found in Patinjavi in 2021. In chlorophyceae, these are the species name, which in Enchistodesmus species, Clostridium species, Pediatum duplex, Pediastrum simplex, Euderina species, Actinastrum hechanki, Syndesmus dimorphus, Syndesmus quadricola, and Monorphidium species. The bold species are found in both the pond. Bacillophyceae, plankton species name in Syndra species, Nichia species, Pleurosigma, Cimbella species, Navicula species, Cytotella species, Frustelia species, Pelorella species, Amphilura species, Cirolilla species, Gamphonema species, Didesmus species, and Urolinia species, Sera granulata species. The bold name which found in both the species are same. In Cyanophyceae, I found Cylindrospermum species, Enabena species, Meristopodia species. In Pathampur pond, Microcystis species, Enabena species, Merismomodia species. In Euglenophyceae, in Thala pond, Euglena species, and Pathampur pond, Euglena species and Pectus species. These are the name I have already told. In the present study, Microcystis eurogynosa were found possibly growing in the water. We supported the study that Microcystis species are most commonly found in eutrophic water of India. Syndesmus species mostly grow abundant in sewage polluted water bodies. Species such as Oscillatoria species, Eugleno species, Syndesmus species, Chlamydomonas species, Navicula species, Chlorella species, Nichesia species, Enkestrodesmus species are well furnished into organically polluted water, sustained by previous study of Ratna Spethiki, Gunale and Balakrishnan, Safari and Gunale and Senapam. These are some photographs of zooplankton species, Keratella filiana, Bronchionus, Ergotholecus foliesa, Copopoda, and Polyelthra vulgaris. These are some photographs of phytoplankton species, Pediastrum species, Syndesmus species, Diatoms, Echinastrums, Ulesoceria species, Monodarphium species. I conclude my research paper here. The quality of pond is important for regulation of biotic and abiotic factor. The present study had provided primary information of plankton diversity of two freshwater body near Dahod. In Thala pond, Rotifera group dominant over other group of zooplankton. Rotifera group is best indicator of eutrophic status of it. This pond may be eutrophic in nature, and this status may be controlling by lesson nutrient and high the fish culture. In Pathampur pond, the Copapoda shows the high diversity. Copapod dominance may suggest the pond may be eutrophicated or oligotrophic in nature. Bacillophyceae being a dominant class followed by chlorophyceae, pseudophyceae, and euglenophyceae needs further study with the climatic factor of the pond to conservation measure. Current study though has been conducted in some selected ponds of Dahod. This will help to get mainly information about plankton diversity, which help for further study related to this area. This is by references. Thank you.
Thank you, ma'am. Our next presentation is Pooja Kamble from Maharshi Dayanand College of Arts, Science and Commerce, Parel. Her topic is synthesis and characterization of silver nanoparticles using shark eye lens extract. Over to you, ma'am.
Thank you so much, ma'am.
5 मिनिटं पण असेल 2 मिनिटं असेल.
Ma'am, I hope I'm audible.
काय बोलले नका.
Yes, ma'am.
कारण मध्ये मध्ये पवाडाचं सारखा ते मागे.
Ma'am, my PPT is visible.
No, ma'am. Share your screen, ma'am.
Ma'am, I have shared my screen.
No. You have not shared yet.
Okay, just a minute. I'll share it once again.
Now it is visible, ma'am.
Okay. Okay, so good afternoon, everyone. I'll start with my presentation of synthesizing and characterization of silver nanoparticles using shark eye lens extract. I am from MD College and I'm working as a research scholar here from Department of Zoology under the guidance of Rishikesh sir. So I'll start this presentation. So basically, we'll start with, ma'am, I'm audible also, right?
Yes, ma'am, you're audible.
So I'll start with an introduction part, like what is nano. So nano term refers to 1 billionth of a meter. So 1 billionth of a meter is known as a nano. And nanotechnology nowadays is one of a vibrant area of research, which is making an impact on all human aspects of human life. So nanoparticles are basically synthesized by using nanotechnology. Nanotechnology is a technology wherein we manipulate the structures based on their atomic, molecular, and macromolecular, and we make use for the betterment of humans. So nanotechnology is basically utilized for making nanoparticles. So nanoparticles have a size which ranges from 1 to 100 nanometer. That means they are so small, and because of their smaller shape and size, they have wider range of applications. So we can synthesize nanoparticles using either physical method, either chemical method, or biological method. Nowadays, we are focusing on biological synthesis of nanoparticles because physical and chemical methods are not environmental friendly as compared to the biological method. So nanoparticles have broader range of applications. That means they are utilized as a catalyst, environmental remediation. Also, they are utilized. They also have antibacterial activity. Vector delivery is also one of an aspect of nanoparticles. So this is about an introductory part of what are nanoparticles. I'm focusing today on green synthesis or biological synthesis of nanoparticle. How green synthesis is done is if a biological material is utilized, and along with that, if we utilize a metal salt, uh, be it silver, be it copper, be it gold, any metal along with metal salt along with a biological material, it is termed as green synthesis, where we are reducing the chemical consequences of it being. So in green synthesis we are utilizing biological material. Why it is favored? Because it is environmentally friendly, it's less toxic, and it's a sustainable approach. Nowadays we are thinking of all the waste material which is available, how to recycle those waste materials. So now our concept is instead of utilizing let's say marine algae or utilizing marine bacteria, the waste which is already generated, can that be recycled and reduced and utilized in a much better way? So nowadays we are recycling, or let's say we are utilizing greener aspects in, we are taking your biological material which are waste material, and we are taking them, and by using them we are utilizing, or we are synthesizing nanoparticles. So my topic is related to marine environment. So marine extracts have not given much importance. They are not utilized more frequently for biosynthesis of nanoparticles. Though they are rich source of reducing agent, they are also used as a capping or a stabilizing agent. So nowadays some research students are focusing on marine extracts. So this slide explains about what is the difference between the physical and the chemical synthesis, and we all know the difference between the physical and the chemical synthesis. Chemical synthesis have more use of toxic chemicals which are environmentally harmful, so people are switching over from chemical and physical synthesis to biological synthesis, which is more environmentally acceptable. So I'll come to my topic of elasmobranch. That means I'm utilizing shark islands. This was suggested by my research guide. Basically, we are working on various different elasmobranch waste which is being generated. Maybe the skin, bones, fins, teeth, whatever elasmobranch waste have been generated, on that we were focusing on. So sir told me to focus on fish islands which have not been so far been investigated. So my topic was to first thing was to get the head from the fish market, shark head from the fish market, which is thrown as a waste. So elasmobranch are not eaten as a delicacy nowadays. They are discarded, or they are uh, used, or let's say they are taken as a bycatch, right? So as the fish production is also increasing, the waste production is also increasing. So to minimize the waste production, what we are doing is whatever the waste is generated, how that can be reduced. So the shark head were bought from the fish market, and then from them the eye were removed from each of the shark, which we get normally from the fish market, and then from that eye the lenses were removed, and how those lenses which have certain amount of proteins can be utilized for production of nanoparticles. The synthesis of nanoparticle is my topic. So how we have utilized the fish waste was the focus here. So the aim is, my present study aims to synthesize nanoparticles from aqueous extract of shark islands. So from shark eye, I have to remove the lenses out, and then the characterization of the nanoparticles which are formed by either SEM or TEM, and also by UV visible spectrophotometer was the aim of my topic. And this started with method and methodology. So after bringing the shark head into the lab, the first step was preparation of the islands extract. So islands extract was done by using the forceps. We removed the eye of shark, and then later on by puncturing the eye we got the lenses, which were transparent round structures which were present. After washing them and heating them in the oven for 1 day for 70 degrees Celsius, what we did was, in the mortar and pestle we crushed those lenses, and we got a powder sort of structure which was weighed, and approximately 15 to 16 shark head were only found on that day, so from the fish market, and it was absolutely free, so we got those. It was only 0.02 gram, the mixture, the powder which was there, it was only 0.02 grams. So I decreased the concentration of distilled water there. Only 20 ml of distilled water was utilized. We boiled the extract on the burner, and then it was filtered with the help of Waterman filter paper, and this is how we got the shark islands extract. After preparing the extract, we had also done for preparation of silver nitrate. So here we are utilizing silver nitrate. You can also utilize different metal salts. I was focusing on silver nitrate. So silver nitrate of 0.01 molar concentration was made, and this is based on the molarity. So 0.0169 gram of silver nitrate was taken and put in a 100 ml of sterile distilled water. All the instruments were autoclaved and used. So once we made this silver nitrate solution, the next step was to check whether there is synthesis of silver nanoparticles. So for this, we took the test tube, set of test tubes, and we made a concentration of, let's say, 10%, 20%, 30%, 40%, and one blank was kept. So in 10%, what we did is we took only 1 ml shark islands extract in it. 9 ml we put distilled water, sterile distilled water, and 10 ml of silver nitrate solution, which was of 0.01 molar. So this was for 10%. So for 20%, 2 ml of islands extract solution, 8 ml of distilled water, and 10 ml of silver nitrate. So this is how the concentration for 30% extract. What we did is 3 ml of the extract islands extract, then 7 ml of distilled water, and the silver nitrate solution, which remained constant for all of the test tubes, like 10 ml. So this was done on the very first day. So we prepared 10%, 20%, 30%, and 40% extract, and one was blank. And in that blank was only 10 ml of distilled water, sterile distilled water, and 10 ml of silver nitrate solution. There was no shark islands extract which was put in the blank. This was just for a reference that whether there is color change or not. So after 3 days, we got some brown precipitate which was seen. And after 3 days, we went for UV visible spectral analysis. We utilized UV visible spectrophotometer of our chemistry lab. And there we checked from, let's say, 340 nanometers till 600 nanometers. We checked for every wavelength whether we are getting a peak or not. So initially for 10, 20, 30 samples, we got a peak at, let's say, 440 nanometers. And then as gradually the concentration increased for 40% extract, we got a peak at 450 nanometers. After confirmation with the guide that there is at least a coloration which is seen and UV visible spectrophotometer readings, then he made us assure that you should go for characterization, and we went for then cement analysis at IQAC lab. I'll show you. So these are the results and discussions. UV readings which was there. So this graph is yellow colored. Peak is for 40%, and rest are like 10, 20, and 30% extract. We got the peak at 440 nanometers. So probably this increase in the extract have probably resulted in the stable nanoparticles, which show the peak at 450 nanometers. Usually nanoparticles are synthesized and which give an OD from 400 to 500 nanometers. So there was a difference here. Some of the samples showed 430 nanometers, whereas if I increase the concentration, it showed the peak at 450 nanometers. So this was the UV visible spectrophotometer analysis. After this, we went for TAME and SAED studies. So transmission electron microscopy was done at IQAC lab, wherein we got specific circular structures. There were also polydispersed, that means there were different shape and size of nanoparticles which were formed, but predominantly we got spherical shaped nanoparticles. Here their sizes was from ranging from 12 nanometer to 67 nanometers. So as the concentration of the extract increases, number of silver nanoparticles which were free, that decreased because they were already there present with the shark islands extract. And in shark islands extract, predominantly there is gamma crystalline protein present. So this was the very first time that instead like a protein and a nanoparticle interaction we could find. There are different amino acids which we want to investigate. Those amino acids are responsible for reducing, capping, and stabilizing the nanoparticles which are formed. So, by using islands, there are different amino acids which are favoring better nanoparticles formation. So, this is what we came across. So, through TM studies, we came across the size of the nanoparticles which are formed. Through area diffraction, we came across that there are certain electrons which are passed through this sample, and through this, if the electrons are passed, then what is the particular size and shape of that particular nanoparticles which is formed is being seen through selected area diffraction. So this too told us that that TM and SAED studies told us that silver nanoparticles are more predominantly spherical in shape. So TM and SAED studies were done. Then SEM, that is scanning electron microscopy, was also done to check the morphology of the silver nanoparticles which are formed. So the morphology, the size was confirmed through TEM. So the SEM tells us what is the morphology of the silver nanoparticle. Are they crystal structure? Are they rod shape? Or which shape are they having? So through SEM, we concluded the quantitative and qualitative analysis of our sample, and through EDX it is confirmed from this graph. It is confirmed that the peak is shown for silver. That means there was silver element or a metal which was present which showed the peak. That means the particles, nanoparticles which are predominantly formed are of silver, not of any other metal. So SEM analysis and EDX was also done to ensure that the nanoparticles are of spherical morphology, and the element which is acting here is silver. So with this short research, we come to a result and conclusion. In this paper, for the very first time, I sir was susceptible whether there will be formation of nanoparticles or not. So initially we just focused on whether the nanoparticles are being formed or not. So we didn't go for a more application part of, let's say, antibacterial activity or any other activity I haven't shown here, because we weren't sure if the nanoparticles will be extracted from the shark islands. So for the very first time, it is being reported, and it is environmental friendly, green, cheap, and simple method for synthesizes of self-assembled nanoparticles. Self-assembled, that means we are not using any external reducing agent here. So itself, the shark islands is having certain amino acids, certain proteins which are helping in reducing and stabilizing the silver particles which are formed. So this method does not require external reducing agent, and through TEM it is observed that the specific size of nanoparticles are formed, which ranges from 12 to 67 nanometers, and we used 10, 20, 30, 40% extract of shark islands, and we also did a confirmatory test with SEM and EDX, and also with a UV visible spectrophotometer, which showed a peak at 440 and 450 nanometers. So the future prospects will be to check the application part of it, that means antibacterial activity, and also to check the proteins which are formed, the composition of the protein, and how to purify those. So that remains as a more things to come. So I think I take this opportunity to thank KLE College for giving me time to present my research. I also thank my guide, Dr. Rishikesh Darvi sir from MD College. And thank you, ma'am. Over to you.
Thank you so much, ma'am.
Stop sharing, ma'am.
Yeah, now we'll take a break of 15 minutes and begin again. Thank you. Pooja ma'am, I think ma'am is sharing her screen. Welcome back. Our next presentation will be by Pooja Patil, and her topic is phytoactives of Moringa oleifera leaves and its medicinal properties, a review. Over to you, Pooja.
Good afternoon to all. Myself Pooja Patil. I am from SYBSC. I am from KLE College, Kalamboli. Today I am going to present my presentation PPT. My topic is phytoactives of Moringa oleifera leaves and its medicinal properties, a review. Let's get started. First of all, introduction. Moringa oleifera leaves are also known as a drumstick tree. It is a native plant from South Africa, Asia, mainly in foothills Himalayas, India. It is very short, easy to cultivate, and it grows quickly. Its leaves are highly nutritious, rich in amino acids, vitamins, minerals, and it has natural antioxidants. This review focuses on the phytoactives and its medicinal properties of these plants. This review divides it into two sections. First section, phytoactives and its roles, and another section, its medicinal properties of these plants. Let's move towards the next slide. Bioactive components in Moringa oleifera and its roles. First, vitamins. The fresh leaves of Moringa oleifera are a good source of vitamin A. It is well established that vitamin A has a important function in vision, reproduction, embryonic growth and development, immunocompetence, and cell differentiation. The fresh leaves of Moringa oleifera are a good source of carotenoids with provitamins A. Moringa oleifera also protect the body from deleterious effects of free radicals, pollutants, and toxins. With it as an antioxidant. Next, polyphenols. The dry leaves of Moringa oleifera, it is a great source of polyphenol compounds such as flavonoids and phenolic acid. The main flavonoids found in Moringa oleifera leaves are milcetin, quercetin, and camphorol. Intake of flavonoids, it has been shown to protect against chronic disease. It is associated with oxidative stress, including cardiovascular disease and cancer. Quercetin. Quercetin, it is found in dry leaves of Moringa oleifera. It is a strong antioxidant having multiple therapeutic properties. It is also having hypolipidemic, hypotensive, and anti-diabetic properties. Next, phenolic acids. Phenolic acid it is a subgroup of phenolic compounds. It is naturally present in plants. These compounds also have antioxidant, anti-inflammatory, anti-mutagenic, and anti-cancer property. Next, chlorogenic acid. Chlorogenic acid has been found lower postprandial blood glucose. It has reduced the glycemic response and has anti-dyslipidemic properties. Next, other phytoactives, alkaloids. Alkaloids are a group of chemical compounds which has contain mostly basic nitrogen atom. Glucosinolates. Glucosinolates are the secondary metabolites in plants. Glucosinolates and isothiocyanate, we both have been found to have important health promoting properties. Tannins. Tannins are water-soluble phenolic compounds. Water-soluble phenolic compound, the concentration of tannins of dry leaves is 13.2 and 20.6 20.6. Tannins must be have to reported that anti-cancer, anti-atherosclerotic, anti-inflammatory, and anti-hepatotoxic properties. Saponins. Moringa oleifera are a good source of saponins. It has the concentration of saponins are 20.6 of freeze and dry leaves of the dry weight. The saponins have anti-cancer properties. Let's see medicinal properties. First is hypolipidemic effects. The phenolic acids and flavonoids has play an important role in lipid regulation. Moringa oleifera also contain beta-sitosterol. It affects cholesterol lowering effect. Saponins. Saponins are found in Moringa oleifera leaf. It has prevents the absorption of cholesterol. Next, antioxidant effect. Due to high concentration of antioxidant present in Moringa oleifera leaves, they have to allow be patient with inflammatory conditions such as cardiovascular disease, cancer, and hypertension. Due to the recent research, children demonstrated with the Moringa oleifera could be an important source of vitamin A. Next, anti-inflammatory and immunomodulatory effect. Extract of Moringa oleifera inhibited human macrophage cytokinin production such as tumor necrosis factor alpha, interleukin 6, and interleukin 8. It has decreased gene expression and production of inflammatory markers. Next, hepatoprotective effect. The methanol extract of Moringa oleifera leaves has a hepatoprotective effect. Moringa oleifera had substantial effect in addition of reduction in lipids and lipid preoxidation levels. And next is anti-hyperglycemic effect, anti-diabetic. Anti-diabetic. Flavonoids and phenolic acid are affects glucose homeostasis, affects glucose homeostasis, including beta cell mass and function. Including beta cell mass and function, it has increased in insulin insensitivity in peripheral tissue. The flavonoids has plays an important role in hyperglycemic action. The group consuming Moringa oleifera extract, it has decreases the monoaldehyde and improves the inflammatory cytokinin. And next, hypotensive hypotensive effect. It has several bioactive compounds. It has isolated four pure more compounds. First is nizanine A, nizanine B, nizamicinin, and fourth is nizanine A plus B. Next, effects on ocular disease. It has major cause of blindness. The Moringa oleifera leaves, pods, and leaf powder. It has also contained high concentration of vitamin A, which can help to prevent night blindness and eye problems. The night blindness and eye problems. It has also consuming oils and leaves improve vitamin A nutrition and delay the development of cataracts. Next is anti-cancer effect. It has been studied for chemopreventive properties. It has also been found inhibit growth of several human cancer. It has the Moringa oleifera leaves also extract proven to be efficient in pancreatic and breast cancer cells. It has also been found including several bioactive compounds. Next, conclusion. As a result of the phytoactive components, prevention of fat accumulation, reduction of insulin resistance, and inflammation. Several animal studies have shown that Moringa oleifera leaves have protective benefits against cardiovascular disease, hypertension, diabetes, NAFLD, Alzheimer's, and other diseases. Additionally, according to certain research, Moringa oleifera leaves chemical composition may potentially offer protection against Parkinson's and Alzheimer's disease. These effects of Moringa oleifera on chronic disease needs to be confirmed by additional human research, including clinical traits. These are the references. Thank you.
Thank you, Pooja Patil. The next presentation will be by Vanchita Matre, and her topic is a review, breast cancer and its contributing factors. Over to you, Vanchita.
A very happy afternoon to each and everyone. Myself, Vanchita Vilas Mhatre from class SYBSc. My topic is breast cancer and its contributing factors. Introduction. Over 7 million people are dead by the breast cancer. Breast cancer is most common among women. It has a great impact on women's life. It affects social, physical, and mental aspects of women. It is most prevalent type of malignant neoplasm.
Vanchita, just 1 minute. Your slide is not moving.
Introduction. More than 7 million people are caused death due to the cancer. Breast cancer. Breast cancer is most common among women. It has a great impact on women's life. It affects social, physical, and mental aspects of women. It is the most prevalent type malignant neoplasm, that is a cancerous tumor. This review deals with several aspects of breast cancer and its contributing elements. Now, moving ahead towards the quality life of a breast cancer patient. WHO defines quality of a life as an individual's view of their place in their life based on their culture and values. Cancer effects on a person's life in a various manner. Mental and emotional effects of illness, stress, pain, economy, etc. are the life quality of the person. Medical professionals can help cancer patients become autonomous in their life affairs. Cancer care aims to improve quality of life and a job capability. Now, what breast cancer exactly means? Breast cancer is the second most cause of death. Most cancerous death among women is from age 45 to 55. Breast cancer requires full tissue resection, chemotherapy, radiation, and hormone treatment. Breast cancer affects the inner layer of milk glands and ducts. Race, economic position, and iodine deficiency are the key factors for the cancer. Now, family and the breast cancer. Breast cancer is a serious threat to women's health. Therapy can affect patients' self-awareness, self-confidence, and self-worth. Mental health issue can affect breast cancer patients. Loss of breast signifies the loss of femininity. Chemotherapy has a major impact on patients' quality of life. As I said, chemotherapy has a major impact. It affects patients' emotional, physical, emotional, and significance of the life. Cancer and its genetic factors. Breast cancer is also caused by hereditary, environmental, and risk factors. Breast cancer cells accumulate genetic and epigenetic alteration. According to the researchers, the main impacts of the risk factor for the breast cancer are age, alcohol, etc. Family history is the most important risk factor for the breast cancer. Breast cancers are linked to the predisposing gene. Weight gain and high calorie intake also contribute to breast cancer development. According to the Copals and Greenwald, breast cancer, high obesity, and BMI are the risk factors for breast cancer in postmenopausal women, not in premenopausal women. BRCA1 and breast cancer. BRCA1 stands for breast cancer gene 1. It is a risk factor for non-genetic breast cancer and most common in menopausal women with help of estrogen receptor. What is the role of estrogen? Estrogen plays in two functions to provide and risk for breast cancer. Estrogen receptor compounds damage DNA. How ovarian cancer is caused? BRCA1 causes the risk of ovarian cancer by 63 to 23% by the age of 50 to 60. If BRCA1 undergoes mutation, it increases the risk of breast cancer. Now, conclusion. It is a major malignant disease affecting all aspects of life. The quest for progenetic biomarker and targets for biological therapeutics has improved breast cancer treatment. Risk factors such as smoking, alcohol use, and diet can be changed, but other factors are fixed. BRCA1 mutation increases the risk of breast cancer. Hormone plays a role in breast cancer, but social and family support can reduce its effect. Here are some references which I took for my paper. Thank you.
Thank you, Vanchika. The next presentation is by Alfia Bakshi, and her topic is Phytoactives of Musa acuminata, pseudostem extract and its medical properties, a review paper. Over to you, Alfia.
A warm good morning to one and a warm good afternoon to one and all present over here. My name is Alfia Bhakshe from SYBSc and my topic is evaluation of phytochemicals and antioxidant activity of Musa acuminata floret extract. So here we begin. Ayurveda stresses the use of plant-based products, that is herbal remedies for good health and treatment of various ailments since ages. Natural compounds produced from plants have been identified with the knowledge of the uses of various plant extracts since ancient times in India. Many medicinal plants have therapeutic effects and can be extracted and used in preparation of drugs. Used directly or in combination with other plant extracts for medication, which is now a common practice all over. Phytomolecules also have played an important role in process of discovering and developing new drugs. They have proved to be very effective in creation of therapeutic interventions for the treatment of wide range of diseases. Musa acuminata is a common plant. Most parts being used, consumed, popularly banana fruit. They are popular because of their nutritive, energy-giving, and medicinal uses. The current study was conducted to investigate the phytochemicals and the free radical scavenging capabilities of Musa acuminata. Next, we have material and methods. First of all, identification and handling of sample. Musa acuminata arid flowers were gathered from Kannur plantation. The plants and flowers were authenticated by normal procedures prior to initiation of study. The harvested flowers were taken to the lab in sterile containers. They were then dried and finely ground, sieved, and powdered dry flowers were kept in non-toxic plastic bags. Next is plant extract preparation. All chemicals were purchased from SE Fine Chemicals, High Media, India. Methanol solvent, that is 200 ml, was used to extract 10 grams of powder bulk. For the extraction procedure, dark maceration for 72 hours at 27 degrees Celsius was adopted and muslin fabric was employed for filtration. After the filtrate was condensed at 45 degrees Celsius, the extract was further used for experimentation. Next, we have qualitative phytochemical screening of crude methanol extract of Musa acuminata was carried out using standard phytochemical procedures. This test conducted for the presence of alkaloids, clobutannins, triterpenoids, flavonoids, lipids, steroids, and terpenoids. Now, what is antioxidants? Antioxidants are a components that reduce the oxidative stress level by prevention of free radicals from damaging DNA, proteins, and lipids. Free radicals are the chemical species which contain one or more unpaired electrons which are highly unstable and cause damage to other molecules by extracting electrons from them in order to attain stability. Next, we have DPPH assay. Unlike laboratory generated free radicals such as hydroxyl radical and superoxide anion, DPPH has an advantage of being unaffected by certain site reactions such as metal ion, chelation, and enzyme inhibition brought about by various additives. A freshly prepared DPPH solution exhibits a deep purple color, which generally fades or disappears when antioxidant is present in medium. Thus, antioxidant molecules can quench DPPH-free radicals by providing hydrogen atom or the electron transfer, conceivably via free radical attack on a DPPH molecule, and convert them to a colorless product. DPPH, that is 2,2-diphenyl 1-picryl hydrazine, or a substituted analogous hydrazine. It results in decrease in absorbance of 518 nanometer, reveals the free radical quenching activity of the plant extract, and the results are expressed as percentage of the ratio of decrease in absorbance at 518 nanometer to the absorbance of DPPH solution activity. Radical scavengers protect tissue from free radicals, thereby preventing several diseases such as cancers. Antioxidant assay, that is DPPH radical scavenging activity. Quantitative measurement of radical scavenging properties of Musa acuminata flower extract was tested using DPPH assay with slight modification. The reaction mixtures contain 50 microliter of test sample using varied concentrations, 80% methanol as planned, plus 5 ml of 0.004% of 2,2-diphenyl 1-picryl hydrazine radical, that is DPPH, scavenging in methanol. The tubes were incubated for 30 minutes in dark room, and the optical density, that is OD, was measured at 517 nm. Color change from violet to yellow indicated the presence of antioxidants. Quantification was calculated by absorbance. Tests were performed in triplicates. DPPH radical scavenging can be expressed in terms of IC50, that is inhibition concentration. The concentration required to achieve 50% scavenging of DPPH radical under experiment condition, IC50 value was calculated from the graph of absorbance at 517 nanometer against methanolic extract concentration and compared with those of standard antioxidant. DPPH radicals concentration was calculated using following equation. DPPH scavenging effect, that is percent inhibition, is equal to AO minus A1 divided by AO into 100%. Whereas AO is absorbance for a control, A1 is absorbance for a sample or standard with the addition of DPPH solution. Here the commercial antioxidant is butylated hydroxytoluene, that is BHT, and the standard antioxidant is absorbic acid. Next, we have statistical analysis. The results were represented as a mean plus minus standard error mean SEM of 3 replicates. The statistical analysis can be significant variations achieved were evaluated by students' tests with probability less than equal to 0.05 treated as significant. Graphs were prepared with probability less than equal to 0.05 treated as significant. Table 1 shows the qualitative phytochemical constituents of Musa acuminata flower extract. The presence of phytochemical constituents are alkaloids, phlobatannins, triterpenoids, flavonoids, and lipids, and the absence are steroids and terpenoids. Next, we have phytoactive constituents and their role. Flavonoids are acknowledged to retain biochemical and pharmacological activities such as anti-inflammatory, antidiuretic, antispasmodic, anti-tumor, anti-allergic, antimicrobial, antifungal, hepatoprotective, and antiviral. The presence of phenolic compounds in extracts is accountable for the antioxidant activity of plant extracts. Alkaloids are connected with therapeutic uses for decades and potentials for disease resistance and stress. Much of the biological properties include analgesic, antispasmodic, antibacterial, cytotoxicity, antiradical, antifungal, anti-inflammatory properties. This table shows the antioxidant activity of methanolic flower extract of Musa acuminata by DPPH assay. The concentration of 0.1 ml shows 23.33% inhibition. The concentration of 0.3 ml shows 28.50% inhibition. Concentration of 1 ml shows 48.30% inhibition. Concentration of 3 ml shows 67.17% inhibition. Concentration of 10 ml shows 96.53% inhibition. The concentration of ascorbic acid shows 0.83 ml, that is 52% inhibition. BHT shows 1 ml concentration, that is 73.14% inhibition. And the total IC50 is 2.55 ml. This graph shows the antioxidant potential of Musa acuminata flower extract, that is DPPH assay. It shows concentration by percent inhibition. Next, we have dietary fiber. These are of two types, soluble and insoluble. It promotes healthy digestion and absorption of food in the gastrointestinal tract. It exerts beneficial role in prevention, management of gut-related problems, cardiovascular diseases, type 2 diabetes, certain types of cancer, and obesity. Flavonoids and tannins, it helps in anti-ulcer and anti-inflammatory activities, and hence could be a wonderful remedy for ulceritis. Phenolics include simple phenols, phenolic acids, that is benzoic and cinnamic acid derivatives, coumarins, flavonoids, stilbenes, hydrosoluble and condensed tannins, lignans, and lignins. It prevents LDL oxidation, hypolipidemic activity, suggesting prevention and treatment of atherosclerosis. Flavonoids and total phenolic compounds shows health food supplement for it helps in head food supplement for breastfeeding mothers, including postpartum mothers and post-cesarean mothers. Unsaturated fatty acids such as oleic, linoleic, and alpha-linoleic acids shows accounting for more than 60% of total fatty acids that may reduce the risk of cardiovascular diseases. The methanolic Musa acuminata flower extract was able to scavenge 97% of DPPH radicals at a level of 10 mg/ml. Methanolic extract of Brax, that is fluoride, exhibited potent DPPH radical scavenging activity even at a lowest stock solution. The table 2 shows that the methanolic extract of Musa acuminata exhibited DPPH scavenging activity with IC50, that is 2.55 mg/ml, attributable to the polar nature of methanol and has been used for the extraction of polar biotic constituents. However, the prospective scavenging activity demonstrated by flower Bract extracts in the current study was low as equated to the positive standard used. Ascorbic acid, that is 0.83 mg/ml, is 52% inhibition. BHT, that is 1 mg/ml, 73% inhibition. The antioxidant action of DPPH assay is connected with the amount of phenolic constituents present in fluoride fractions, which necessitates screening through other battery of antioxidant assay like ferric reducing FRAP assay, Prolong's equivalent antioxidant capacity TAAC or ABTS assay, copper reduction cuprate assay, and reducing power assay, that is RP. Hydrogen atom transfer reaction assay include crossing bleaching assay, the total peroxyl radical trapping antioxidant parameter, that is TRAP assay, total oxyradical scavenging capacity TOSC assay, and oxygen radical absorbance capacity, that is ORAC assay. The traditional uses of banana flour have low glycemic index, diabetic symptoms like hyperglycemia, polyuria, polyphagia, polydipsia, urine sugar, and body weight reduce. Banana blossoms helps to treat excessive blood loss during the menstrual cycle. Consumption of 1 cooked banana flour with a cup of curd is most effective treatment for excessive bleeding during menstruation, which increases the level of progesterone, thus reduces bleeding associated with menorrhagia. Traditionally, banana flour is used to treat women who are suffering from polycystic ovarian syndrome, that is POS. Next, we have conclusion. Plants have contributed to a lot of medicinal compounds being used today to treat diseases like cancer, jaundice, diabetes, inflammation, etc. They are vital sources of wide variety of phytochemicals from which novel therapeutic agents can be discovered. In this view, attempts were made in the present study to evaluate the phytochemical components and antioxidant activity of methanolic extract of Musa acuminata flowers in vitro employing the simple DPPH assay. The present study is a preliminary evaluation of activity of Musa acuminata which provides a scientific validation for the use of this plant for treatment of plethora of diseases in flock fraud medicine. Further research work to analyze antioxidant potential is necessitated and determine other varied activities of Musa acuminata in vitro and in vivo which are necessary to potentiate the uses of plant. These are some of the references. Thank you.
Thank you, Alfia. The next presentation will be by Vanchita, and her topic is a survey on knowledge and awareness of anemia among female undergraduate students of Navi Mumbai. Over to you, Vanchita.
Very good afternoon to everyone. Myself Anshita Vilas Mathri from class SYBSc. My topic is a survey on knowledge and awareness of anemia among female undergraduate students of Navi Mumbai. Heading towards the introduction, anemia is a global health problem. It is a condition in which there is a deficiency of RBCs or hemoglobin in the blood resulting in pallor and weakness. It occurs in all stages of life cycle, but it is more prevalent in pregnant women and young children. It has an iron deficiency anemia. IDA was considered to be the most important contributing factors to the global burden of the disease. Nutritional anemia is more likely to occur in adolescents. Undergraduate students are vulnerable population who experience anemia. Anemia is particularly due to iron deficiency during adolescence. It may impair physical and mental development as well as the behavioral and cognitive development. It has a risk factors which includes low intake and poor absorption of iron from diet high in phytate. Phytates are the insoluble complex which are present in plants or phenolic compounds and a period of life where iron requirements are especially high. The main objective of doing this survey was to assess the knowledge and awareness about anemia among female undergraduate students. Its measure was to prevalence the anemia among undergraduate students. It compared the socio-demographic factors related to anemia and its correlation. Poverty, limited education and access to knowledge, inadequate dietary and nutrition intake, and infectious disease are suggested to contribute to anemia as well as co-occurrence of stunning. Now moving ahead towards materials and methods, study design and participants. There were total 112 undergraduate girls aged from 18 to 21 who lived in Navi Mumbai, Maharashtra participated in the study. The questionnaire had two sections. Section 1 dealt with the question related to socio-demographic factors that influenced adolescents. Knowledge toward anemia included name, resident, age, course of study, institution, state, family type, education of parents, and occupation of the head of the home. Section 2 included 13 variables consistent of knowledge and awareness components which addressed various aspects related to anemia. Association between awareness, anemia had other factors which assessed. The correct answers was marked as aware and incorrect as unaware, and data was converted into the percentage. The complete data was collected, entered in Excel sheet, and the results were analyzed. Here are the questionnaires. The total components of knowledge and awareness consist of 13 variables. Socio-demographic factors, access to knowledge, attitude, practice, that is KAP. Source of information is also acknowledged as a factor to anemia. As the proximal cause of iron and other micronutrient deficiency, these factors can have an impact on the quality of food, access to the nutrient-rich diets, health, cleanliness, sanitization, and underlying disorders. Anemia is a risk factor for defects in iron and other micronutrients. Here is the result and discussion. Nearly half of the adults and girls were anemic, that is 47%. About 60% of the adults and girls had heard about anemia. 45% knew that there is a decrease in RBC's count or an hemoglobin concentration. 39.6% of responders had knowledge of the effect of anemia. Almost all of those who had heard of anemia knew at least one symptom. Some were also able to identify the cause of anemia and its prevention. However, fewer girls, that is 20%, knew of the knowledge of foods that worsen anemia. 46.6% of girls were aware of the prevention of anemia. Almost 60% of adults and girls use masks and social media as the source of anemia information. A few were informed about anemia by college and health staff, that is 24.1%. 15.6% informed by their families and their peers. 47% of anemic responders, 38% came from the nuclear family in the comparison to extent or a joint family, indicating a correlation. There seems to be an impact of mothers' education in their children's knowledge, awareness, and incidence to anemia. Similarly, there is a correlation of incidence of anemia, that is 46.4%, with that of nature of job of households occupation, that is non-permanent or not working, and that of responders who were anemic. Conclusion. This study reflects that the basic knowledge of anemia and proper information pertaining to the condition, effects, and treatment of anemia is lacking in undergraduate students. Strategy to reduce the risk of anemia in this population should combine KAP improvement with other known effective nutrition intervention, that is, example, micronutrient supplementation. Public health and nutrition education should integrate aspects that could contribute to anemia prevention. This study highly suggests the provision of health education can be implementation of educational, awareness, screening and nutritional programs, periodic educational interventions such as seminars, health checkup is warranted with special reference to the adolescent age group. Here are the references. Thank you.
Thank you, Vanchita. The next presentation is by Alfia Bakshi again, and her topic is evaluation of phytochemicals and antioxidant activity of Musa acuminata floret extract. Over to you.
A very good afternoon to one and all present. Myself Alfia Bhakshe from SYBSC, and my topic for today is phytoactives of Musa acuminata pseudostem extract and its medicinal properties, a review. First of all, introduction. Musa acuminata, it is also known as Cavendish banana or banana. It is a species of banana native to southern Asia. Its range comprising from Indian subcontinent and Southeast Asia. Many of the modern edible desert bananas are from this species. The trunk, known as sheets, emerging from completely or partially buried combs. This review focuses on pseudostem. It is made up of tightly packed layers of leaf phytoactives and medicinal property of this plant. This review comprises of two parts, that is phytoactives and the medicinal properties. Bioactive components in Musa acuminata contains a lot of nutrients and minerals that are beneficial to health. Its vitamin C content is 15%, which is regarded as a familiar antioxidant. It is implicated in the causation of many disorders like arthritis, diabetes, arteriosclerosis, age-related macular degeneration, certain types of cancer, inflammation, genotoxicity, and Alzheimer's disease. The other phytoactives are the native banana pseudostem flower, that is NBPF, and tender core of pseudostem flower, that is TCBPF, for chemical and functional properties. Its higher content of polyphenols, flavonoids, total dietary fiber, insoluble dietary fiber, lignin, hemicellulose, cellulose, antioxidant capacity, and free radical scavenging capacity in NBPF and then TCBPF. It's rich in many bioactive compounds such as carotenoids, flavonoids, phenolics, amines, vitamin C, vitamin E, having antioxidant activity to provide many human health benefits. It explains the use of this plant for treatment of different ailments or diseases. Medicinal properties. It is used in treatment of various diseases such as fever, cough, bronchitis, dysentery, allergic infections, sexually transmitted infections, and some of the non-communicable diseases. It helps in weight loss. It is used to help in weight loss due to high fiber content, which keeps you full for a long time. It helps to treat urinary tract infections, as it is a diuretic that aids in removal of toxins and the cleansing of urinary tract. It helps in treating anemia. It is used to cure anemia due to its high level of vitamin B6 and iron. It aids in production of hemoglobin in the blood. It is good for diabetic patients, as it has a low glycemic index and quite an important factor for any diabetic patient. It helps in burning belly fat. It aids in digestion and metabolism of a body. It is high in fiber, fills your stomach, and keeps you satiated for a longer period of time. It helps in asthma relief, as it is considered to have antipyretic properties beneficial for disease of respiratory system. It also helps in protecting neuronal cells for oxidative stress induced neurotoxicity. It reduces risk of oxidative stress induced neurodegenerative disease like Alzheimer's disease. Conclusion. Musa acuminata pseudostem has been used by indigenous people to treat and heal many infections and diseases, and many models have provided evidence of efficiency. It possesses pharmacological properties and can be used in designing potent therapeutic agents. Pharmacological studies support the traditional uses of the plant, and the animal model provide evidence of efficiency. These are some of the references. Thank you.
Thank you, Alfia. The next presentation is by Amisha Yadav, and her topic is Effect of Music Therapy on Mental Health.
Good afternoon, everyone. My name is Amisha Yadav. I am from class FYBSc. My research paper topic is Effect of Music Therapy on Mental Health. So this is the content. It includes abstract, introduction, objective, hypothesis, material and methods, result, discussion, conclusion, and references. Moving towards the introduction. So what is the music? Music is the language of the soul. It's an art of producing pleasing or expressive combination of tones, especially with melody, rhythm, and usually harmony. People have always found music significant in their lives, whether for enjoyment in always listening, performing, or composing music. Your entire brain can be activated when you listen to music. Music may cause you to feel joyful, sad, angry, hyper, relaxed, etc. And sometimes you can feel more than one emotion while hearing a song. Music therapy is a type of expressive arts that uses music to improve and maintain the psychological, physical, and social well-being of individuals. Music therapy is an effective component of the treatment of depression, insomnia, and can help in pain management. Music can activate regions of the brain that influence things like memory, emotions, movement, sensory relay, some involuntary functions, and decision making. It also strengthens motor skills and brings improvement in communication for children and young adults who face learning disabilities. Use of music therapy releases endorphins, which helps in relieving stress and pain. So, objective of my research paper is to find the effect of music therapy on the mental health of an individual, to evaluate positive effects of music on mental health, to highlight the importance of music being used as a medicine. um, Null hypothesis is music therapy has no positive effect on mental health, while the alternate hypothesis is music therapy has a positive effect on mental health. Material and methods that I have used for my research paper. I have used secondary data from effects of music inquiry bibli.com. It includes the following these techniques. First is vibroacoustic therapy technique, and second is Mozart music technique. In the vibroacoustic therapy technique, it involves the user lying on a mat, lying on a bed, or sitting on a chair equipped with speakers that send out vibrations that can be both heard and felt at precise computer generated frequencies. According to another research, Mozart music was used to study the effects on the growth of premature babies. So result shows that music is an excellent healing tool for the brain because it activates so many parts of it, which truly have a good effect on a person's mental health. Music therapy has brought a huge impact on people's psychological health, enabling patients to suffer less pain so that they can live without stress and anxiety. Music is truly amazing. According to the studies, patients diagnosed with the mental disorders have shown a visible improvement in their mental health after using music therapy. Overall, music therapy can increase positive feelings. Discussion says that vibroacoustic therapy is a good example of music therapy being used as a medicine. In this method, sound of low frequency is used to make vibrations, which is then applied straight to the body. During this therapy, the patient sits on a chair or lies on a mat or bed embedded with speakers that transmit vibrations at specific computer generated frequencies that can be heard and felt. It is found that short-term use of this vibroacoustic therapy with Parkinson's disease patients led to improvement in symptoms, including less rigidity, and the patients began to walk with a good speed and by taking bigger steps and also with less tremor. One more study was conducted by Dr. Drod Mendel and Dr. Ronit Lubetsky at Tel Aviv University. They played half an hour of Mozart every day to premature babies and got amazing results. The babies who listened to the music grew much faster than those who weren't listening to the music. The researchers still aren't sure what is causing the effect, but they think it has to do with the calming effects of classical music, which can help reduce stress and boost the immune system of listeners even as young as babies. Research shows that there can be improvement in the activity of the neural system if the musical training is given to the child. So overall conclusion of my research paper is that music therapy is found to be effective on a person's mental health. So the present study favors H1, which is alternate hypothesis, according to which music therapy has a positive effect on mental health. These are the references which I have used for my study or for my research paper. Thank you.
Thank you, Amisha. The next presentation The next presentation is by Ujala Yadav, and her topic is the study of difference between wings of moth and butterfly. Over to Ujala.
Good afternoon to all. Myself Ujala Yadav, FI BSc. My research topic is study the difference between wings of moth and butterfly. The content of my presentation: introduction, objectives, hypothesis, material and method, result and discussion, conclusion, references. Introduction. The order Lepidoptera include insects like butterfly and moth, which are well-known and cavity insects. Examining the antennae is one of the simplest methods to distinguish between a butterfly and a moth. The antenna of a butterfly have a club-like form, a long stem, and a bulbous tip, while antenna of a moth have storage or feather. Moth and butterfly both have scale covering their body and wings, which is one of their unique similarity. Butterfly frequently fold their wings over their back in a vertical fashion, and moth frequently hold their wings in shape of tent to conceal their abdomen. The wings and pattern of butterfly and frequently wider and more colorful. Moth often have smaller wings than are dull in color. Objectives: to study the difference between the moth and butterfly, to study the difference in wings of moth and butterfly, to study the difference in life cycle of moth and butterfly. Hypothesis: null hypothesis, there is no difference between the wings of moth and a butterfly. Alternate hypothesis, there is difference between the wings of moth and butterfly. Material and method: secondary data was collected and analyzed. Result and discussion: it is observed that butterfly have long, narrow antenna and moth have thick, feathery antenna. Moth luster, whereas butterfly have vivid color. Their wings have many colors and patterns that serve important communication purpose in addition to being purely ornamental. By serving as collab warming possible predator, that butterfly body has toxic or luring potential make that pattern on butterfly wings can consist them to avoid being eaten by human or other animal. It is believed that these two species did not give birth to their offspring simultaneously. Some biologists believe that butterfly consist from various species of moth. Butterfly frequently their wings over their back in a vertical fashion. Moth frequently hold their wings in shape of tent to conceal their abdomen. Butterfly typically have larger wings and more colorful patterning. Moth frequently have more compact dull color wings. A frenulum which connect the wings is a feature of moth. Frenulum are not present in butterfly. In order for the wings to function together during flight, frenulum connect the forearm to hind wings. Majority of time, butterfly only fly during day. Moth typically fly at night, are a not nocturnal creature. However, certain butterfly have crepuscular, which means they only fly and drawn to night. To other moth like buckmouth are nocturnal. This is the image of stages of life cycle of moth and butterfly. Conclusion: all members of Lepidoptera order, which include butterfly and moth, undergo a four-stage life cycle. Each stage of life, insect's life, including egg, larva, pupa, and adult, has a specific function. This is the some references. Thank you.
Thank you, Ujala. The next presentation is by Avinash Chirsagar, and his topic is Study of Water Quality and its Effect on Residents in Navi Mumbai, India. Over to you, Avinash.
Good afternoon, everyone. Myself, Avinash Sagar from class first year BSc. The topic of my research paper is Study of Water Quality and its Effects on Resident in Navi Mumbai, India. So the content of research paper is abstract, objective, hypothesis, water quality and its effect on residents, conclusion and references. So as we move, abstract is in order to establish industries, IT parks, office spaces and housing facilities, Navi Mumbai, a rapidly expanding industrial city in Maharashtra, was developed in the 1970s as a satellite city of Mumbai. Dams provide the water for Navi Mumbai, but urban settlements were not connected to the city's water supply until 2015. They had to rely on municipal taps or tankers to meet their needs. The researchers have studied the effectiveness of the links as well as the impact on the region's health problem. Water is yet a scarce resource and its accessibility and effectiveness is yet questionable. The, the equitable supply of fresh drinking water is still not fully operational in Navi Mumbai. The study observed that drinking water supplies through pipeline in urban areas of Navi Mumbai, but 24 into 7 supply is still a challenge that is mandatory to maintain water quality as well as prerequisite as per sustainable development goals. A NMMC report on water quality was used for the study. The objective of paper is to study the water quality in Navi Mumbai and to know the factors affecting the water quality. Null hypothesis, there is no effect of water quality on resident in Navi Mumbai, India. And the alternative hypothesis is an effect of water quality on resident in Navi Mumbai, India. Material and methods. Secondary data was collected from the official website of a NMMC academic year 2018-19. The table number 1 shows the annual average water quality of lakes in NMMC area in 2018-19. It shows the cities in Navi Mumbai and the lakes in the cities, which shows the pH level, suspended solid level, TDS level, DO level, BOD level, and COD level. The next table shows the person affected by waterborne diseases in NMMC in year 2018-19, which shows the Airoli has the highest person affected by the waterborne disease, that is 86 in the year 2011-12. As the academic year passes, it shows low in the diseases. This is the graphical presentation of the previous table that shows the patient per node in 2011-12 has the highest in Airoli. The result shows that waterborne diseases, water contamination by pathogenic bacteria, viruses, or protozoa is the cause of waterborne illness. When individuals use water for drinking, cooking, or other domestic or recreational activities, these chemicals are transferred to them. When such dangerous bacteria contaminate drinking water, either at the source through seepage of contaminated runoff water or inside the pipe distribution network, an individual health is jeopardized. In addition, dirty water handling during transportation or at home might contaminate clean water. The health department at NMMC has taken aggressive steps to stop the spread of this illness, and its devoted qualified staff has been working relentlessly to mitigate this illness through its yearly preventive efforts. The drinking water quality at the tap end. Before being supplied and distributed to the NMMC area, the Morbe Dam, the area's main source of drinking water, is treated at the Bhokarpada Water Treatment Plant. In three categories, domestic, commercial, Gauthan, and slums, define the city's areas. The potability of the drinking water is continuously checked and monitored by NMMC at several locations throughout the city. The firm took preventive precautions in case there is any contamination. A total of 20,376 samples were examined during the year 2018-19, and 5.17% non-potable samples, that is 1,053, were found in the total samples. It is concluded that the water quality has negatively affected the health of citizens in Navi Mumbai to a great extent, especially in Airoli in 2011-12. But later it was minimized with the efforts of NMMC through various programs. These are the references we took for our research. Thank you.
Thank you, Avinash. The next presentation is by Kashish Ojha, and her topic is Study of Bird Diversity and Mangroves in Navi Mumbai, India. Over to you, Kashish.
Good afternoon to all. My name is Kashish Ojha from class FY My presentation topic is Study of Bird Biodiversity and Mangrove in Navi Mumbai, India. Content for my topic is Introduction, Objective, Hypothesis, Material and Method, Result and Discussion, Conclusion, and References. So let's begin with introduction. The Indian subcontinent reported 1,313 different species of birds. BirdLife International designated this area as an endemic bird area in recognition of its remarkable bird diversity and local endemism. Pawar et al. In 2020, over the course of a year, used point count to method to survey birds in various locations at dawn and dusk from June 2019 to May 2020. A wide variety of birds were seen, including 102 species from 16 orders, 48 families, and 84 genera. Prabhakar Pawar in 2011 recorded that mangroves of the Uran coast are home to 56 species of birds spanning 29 families, 11 orders, and 46 genera. Of the species that were recorded belonging to the order Passeriformes, Sisoniformes, Cedriformes, Anseriformes, Coraciformes, Falsoniformes, order Columbiformes, Gruiformes, and order Cuculiformes. Objectives are to understand the variability of bird occurrences in natural situations, to comprehend how disturbance and habitat alternation affect bird communities, to learn more about how birds keep an ecosystem in balance. Hypothesis. Null hypothesis says that there is no bird biodiversity with respect to mangroves in Navi Mumbai, India. Alternate hypothesis says there is bird biodiversity with respect to mangroves in Navi Mumbai. Material and method. We research secondary data and utilize it for our studies. From the literature review, it is clear that a wide variety of birds were seen, including 102 species from 16 orders, 48 families, and 84 genera. As can be seen in Table 1, the Passeriform family includes 45 species, which is dominating over the other, while Cuculiform, Galliform, Pinicoptiferiform, Psittaciform, and Suliform shows 1 species each other, which is least in number. In Table 2, a total of 20 species of true mangroves and mangrove associated belonging to 17 genera, 13 families, and 13 orders were recorded. There are total of 8 species of real mangroves, which are found in 6 genera, 4 families, and 4 orders. Mangrove associate were represented by 12 species, 11 genera, 9 families, and 9 orders. Here are Here are some glimpses of bird diversity like Accipital batida, Ardea alba, Tyto alba, and many more seen in Navi Mumbai. Conclusion. It is important to take into account additional potential factors that could have a stronger impact on bird species composition and diversity in order to gain a better understanding of this topic. Birds play a significant role in sustaining biodiversity since they are keystone species that ensure the survival and reproduction of a variety of other species. Therefore, the loss of birds could have a significant impact on the environment and reduce biodiversity. So, present analysis favors an alternate hypothesis which states that there is significant relationship between biodiversity and mangroves in Navi Mumbai, India. These are some references which I have taken for my study. Thank you.
Thank you, Kashish. The next presentation is by Pooja Patil, and her topic is a review on physico-chemical properties of water. Over to you, Pooja.
Good afternoon to all. Myself Pooja Patil. Today I'm going to present my presentation PPT. My topic is a review on physico-chemical properties of water. Let's get started. First of all, introduction. Water. Water it is essential for life and development of all living things on the earth. As a result, human activity, industrialization, use of fertilizer in agriculture, and rising human population, the environment is severely contaminated. In order to avoid infection and enhance quality of life, it is essential to have access to clean water. Groundwater quality, it is determined by a variety of chemical components and their concentration. Industrial and municipal solid waste, it is one of the main contributors to surface groundwater pollution. Next, physicochemical parameters. The variety of physicochemical parameters, it must be used to test water. In excess, we need its quality and purity. The several kinds of floating, dissolving, suspended microbiological and bacteriological pollutants are present in water. Then physical test, it has carried out to evaluate the substances, physical characteristics such as temperature, color, odor, pH, turbidity, TDS, etc. Then chemical test, it has carried out to evaluate the substances BOD, COD, dissolved oxygen content, alkalinity, hardness, and other characteristics. In the water quality, it has responded to determine trace metal, heavy metals, and organic pesticide residue in order to acquire higher quality and purity. Temperature. In a stable system, the water temperature controls frequency of all chemical reaction. Then pH. pH plays an important role to determine toxicity of water. The total alkalinity and electrical conductivity had a positive correlation with pH. The more toxic water is correspond to lower pH value. During lower oxygen correspond with high temperature, the pH decrease chlorophyll content. Next is electrical conductivity. There are 10 characteristics: pH, temperature, alkalinity, TDS, BOD, COD. It has a 10 characteristics correlated with conductivity. It has managing the conductivity of water. It has regulates water quality and check underground drinking of water. Next, carbon dioxide. The most significant greenhouse gas on the earth is CO2. All aquatic ecosystem produces carbon dioxide as the byproduct of organic compound or organic carbon. Next is alkalinity. It has served as a pH stabilizer, mostly composed of bicarbonate and carbonate. The main cause of alkalinity in boiler water of hydroxyl and carbonate ions. Dissolved oxygen. One of the most important parameter is dissolved oxygen. It has connection to water, provides both direct and indirect information such as stratification, bacterial activity, and photosynthesis. As the summer progresses, the temperature of antimicrobial activity, Next is biochemical oxygen demand. Water contamination is determined by BOD, which is expressed in milligram per liter. The amount of dissolved oxygen required for the metabolic breakdown of organic molecules and the oxidation of some inorganic elements is known as BOD. Example, iron and sulfides. Usually the BOD is performed 5 days course. Next is chemical oxygen demand, COD. Another indicator of organic material contamination in water is COD, which is measured in milligram per liter. The concentration of dissolved oxygen is the quantity required to chemically oxidize the BOD organic material in water. The BOD and COD has critical factor to the environmental health of a surface water supply. Next, carbonate. The presence of carbonate is indicated if the pH reaches 8.3. It has determined the titration of phenolphthalein and calibrated with hydrochloric acid. Next is bicarbonate. Methyl orange is used as an indicator during a titration with standardized hydrochloric acid to measure it. The pH below the pH below the 4.0, the methyl orange turns yellow. The carbonic acid breaks down this pH, produce carbon dioxide and water. Next is some physical analysis study of polluted water sample in India. The physicochemical parameter study, it is important to assess water quality. The physicochemical parameters were found to exceed permissible limit of CPCB, Central Polluted Control Board. Dilution during rainy season decreases metal concentration, but biomagnification and bioaccumulation increase it. Non-polluted water can be used for domestic and irrigation, but highly impure water has various effects on humans, domestic purpose and industrial use. Next is water quality parameter of groundwater sample from study area and comparison with permissible values. Water must be tested with different physicochemical parameters to determine its quality and purity. The physical tests such as temperature, color, odor, pH, turbidity, and TDS, etc. The chemical tests such as BOD, COD, dissolved oxygen, alkalinity, hardness, and other characters should be performed to determine its trace metal, heavy metal content with organic residue. Only in developed countries this criteria is monitored. Next, conclusion. It is important to test water before it is used for drinking, domestic, agriculture, and industrial purpose. Groundwater is the most important source of water supply for drinking, irrigation, and industrial purposes, but its quality cannot be restored by stopping the pollutants from the source. Therefore, it is important to regularly monitor the quality of groundwater and devise ways to means to protect it. These are the references, and thank you.
Thank you, Pooja.
Very happy afternoon.
Okay, the next presentation is by Vanchita Matre again, and her topic is Impact of Global Climate Change and Sustainable Mitigation Measures, a review paper. Over to you, Vanchita.
A very happy afternoon to everyone. Myself, Anchita Vilas Matre from class SYBSC. My topic is Impact of Global Climate Change and Sustainable Mitigation Measure, a review. So, the list of content which my topic includes are introduction, objective, hypothesis, impact of global climate change and sustainable mitigation measures, conclusion and references. Now, introduction. The global changes have been seen from last 65 years. Globally, climate change is a difficult worldwide challenge that has impact on several biological,
environmental, socio-political, and socio-economic decisions. The comprehensive long-term patterns in temperature and precipitation, as well as other factors like atmospheric pressure and humidity, are used to define climate change. The main objective to study this paper was to understand the impact of climate change in worldwide, to comprehend how climate change can affect different sectors worldwide, to learn more about how to maintain the climate change, and to study how to deal with climatic change on human health, economic sectors, etc. Hypothesis. The null hypothesis says that there is no impact of climate change worldwide, and the alternate hypothesis says that there is an impact of climate change worldwide. Impact of global climate change and sustainable mitigation include some measures. They are as follows. Climate change. Climate change has a negative impact on the human health. Climate change and antimicrobial resistance with corresponding economic cost. It has an psychological impacts of climate change. Psychological impacts of climate change are greatly influenced by the weaker section. Climate change impact on tourism. It is the most important source of income, wealth development, regenerating spectacular foreign exchange, and tool for entrepreneurs, etc. Climate change impacts on economic sectors. Climate change impacts on economic sectors. Climate change impacts on forest. Conclusion. Climate change is causing increase in global temperature, carbon dioxide, and precipitation. Climate change can create a suitable environment for invasive species to colonize and spread, threatening the economy or environment. Climate change impacts global forests, food security, and psychological well-being, requiring mitigation and adaptation strategies. Here are some references. Thank you.
Thank you, Vanchika. The next presentation is by Alpia Bakshi, and her topic is Impact of Junk Food and Fast Food Consumption Among Adolescents in Kalamboli, Navi Mumbai, India. Over to you, Alpia.
Thank you, ma'am. Good afternoon, one and all present over here. My name is Alfia Bhakshi from SYBSC, and my today's topic is Impact of Junk Food and Fast Food Consumption Among Adolescents in Kalamboli, Navi Mumbai, India. First of all, what is junk food? It refers to the food that contribute lots of calories but little nutritional value. It is also known as discretionary food or optional food. Junk food falls into categories of snack food or fast food. Snack food like chips, cheese puffs, candy bars, snack cakes, and cookies. It doesn't play a role in healthy eating, especially if it's eaten to excess. Objectives of junk food are to understand the choices of adolescents about junk food and fast food, to know the consumption pattern of junk food and fast food by adolescents, to analyze the health effects caused by junk food and fast food. The hypotheses of junk foods are: null hypothesis says there is no impact of junk food and fast food consumption among adolescents. Alternate hypothesis says there is impact of junk food and fast food consumption among adolescents. Material and methods. A self-administered questionnaire was used for data collection from the adolescents. Next is result. In table 1, it shows the age group of adolescents on impact of junk food and fast food consumption in Kalamboli, Navi Mumbai. The age group of 17 to 18 years shows 109 number of response. The age group of 19 to 20 years shows 326 number of responses. The age group of 21 to 22 years shows 87% responses. The 19 to 20 years old shows the maximum number of responses. Table 2 shows gender of adolescent about junk food and fast food consumption in Kalamboli, Navi Mumbai, India. There are 250 number of responses in female and 272 in males. This Pie diagram shows the number of responses of figure 1 and figure 2. In figure 1, it shows age group of adolescent on impact of junk food and fast food consumption in Kalamboli, Navi Mumbai. 19 to 20 years shows 63%. 17 to 18 years shows 21%, and the lowest is 21 to 22 years shows 17%. In figure 2, it shows the gender of adolescent about junk food and fast food consumption in Kalamboli, Navi Mumbai. In female, it shows 48%, and in male, it shows 52%. Table 3 shows yes and no responses of adolescent on impact of junk food and fast food consumption in Kalamboli, Navi Mumbai, India. 78.2% response says 78.2% adolescents says that they eat junk food. 21.8% adolescents says they do not eat junk food. 10.8% response says they do have health issues. 89.2% response says they do not have any health issues. 21.8% adolescents says they do have peer influence, whereas 78.2% adolescents says they do not have any peer influence on them. 40.8% responses says that parents allow to consume junk food, whereas 58.6% responses says that parents do not allow them to to consume junk food. 52% adolescents says they do regular exercise, and 48% adolescents says they do not do regular exercise. This is the bar diagram that shows the yes and no response of adolescent on impact of junk food and fast food consumption in Kalamboli, Navi Mumbai. Next table shows choices of adolescent on impact of junk food and fast food consumption in Kalamboli, Navi Mumbai. 391 adolescents says they eat salty snacks, whereas 109 eats sweets. 76 adolescents says they eat daily junk food. 196 says occasionally, and 228 says thrice a week. 326 adolescents they have healthy life, whereas 120 says lean and thin, and 54 says overweight. This is a bar diagram that shows the choices of adolescents about junk food and fast food consumption in Kalamboli, Navi Mumbai. That is what type of junk food they eat, how frequently they eat junk food, and what type of health they have. Next is junk food consumption has been linked to an increased incidence of adolescent obesity and overweight. Thus, proper measures must be taken to decrease consumption of such foods. Teenagers are more prone than adults to eat junk food at homes, at schools, in restaurants, and while traveling, according to some researchers. Another study shows that people who eat lunch at schools, hotels, and bakeries are more likely to consume junk food. Eating out for supper, eating vegetables and fruits, parental influence on eating patterns have all been linked to lower junk food intake. Participants were more likely to lack adequate knowledge about junk food and its long-term effects on public health. We recorded that maximum parents do not allow their children to eat junk food and fast food since they know the impact of it, but children may be lacking the knowledge regarding this. Similar findings were recorded. These are some facts to remember. Fan of junk food? There is no need to despair. Fine to eat junk food if you eat them in moderation. If you are eating a well-balanced diet overall and maintaining a healthy weight, you shouldn't have any problem eating junk food occasionally. But just remember, your body needs a fuel in the form of nutrients to perform at its best. So when you replace nutrient-rich foods with high-calorie or low-nutrient snacks, your body doesn't get the fuel it needs. In conclusion, according to the result of a study, adolescents are eating more junk food, which may be factor in their poor growth, consumption on transit, dining out. Also, it was discovered that the individual's increased consumption of junk food was more influenced by their family and peer relationships. Consumption of junk food invites various health problems. The main problem with junk food are increasing childhood obesity, which further leads many health complications in children. The attracting advertisements are largely responsible for the mind wake-up of children to consume junk food. Multinational companies are attracting the new customers by attractive and aggressive marketing strategies. Parents should take care of eating habit of their children and protect them from junk food by increasing the awareness about the health problems associated with junk food and fast food. The habit of junk food can be avoided by strong willpower and awareness of side effects associated with them. School or college canteen also ensure to keep healthy food in their menu rather than focusing on only junk food. Children must be educated about the harmful effects of junk food on health. This may be pretty helpful in avoiding of junk food and problem associated with them. These are some of the references. Thank you.
Thank you, Alfia. The next presentation and the last one is by Prachi Patil, and her topic is Study of Genetic Traits of Rolling Tongue and its relation with blood group in India. Over to you, Prachi.
Good afternoon, everyone. Myself is Prachi Patil, and I'm from SYBSC. My topic is Study of Genetic Traits, Rolling Tongue and its relation with blood group in India. Introduction. By inheritance, genetic information is passed from parents to child. Because of this, family members frequently have traits in common. Morphogenetic traits such as facial shape, hair color, eye color, and disease are physical characteristics that can be passed from parent to child. The tongue is a flexible organ with many different shapes and forms that is formed of muscles, tissue, and covered in mucosa membrane. The ability to roll the tongue lateral edges up and into a tube is known as tongue rolling. The method of passing biological qualities from parents to children through gene inheritance is called as inheritance. Objectives: to study the genetic traits of rolling tongue and its relation with blood group in India, to identify the relation between blood group and its effect on rolling tongue. Hypothesis. Null hypothesis. There is no relation of rolling tongue with blood group in India. Alternate hypothesis. There is relation of rolling tongue with blood group in India. Material and methods. Secondary data was obtained from the given link and used for study. Results. Table and figure 1 indicates inheritance of rolling and non-rolling of tongue from parents to offspring. The given graph shows the data that is presented over here. It indicates when both the parents have rolling tongue, 28 of the offspring shows rolling tongue, while 5 of the offspring shows non-rolling tongue. When one of the parents has rolling tongue and the another has non-rolling tongue, 33 of the offspring shows rolling tongue, while 22 of the offspring shows non-rolling tongue. When both the parents have rolling tongue, non-rolling tongue, 4 of the offspring shows rolling tongue, while 9 of the offspring shows non-rolling tongue. The table 2 and graph indicates inheritance of rolling and non-rolling of tongue from parents to offspring. Here, the data indicates that when both the parents have rolling tongue, 90% of the offspring produced shows rolling tongue. When one of the parents has non-rolling tongue and the another has rolling tongue, 68% of the offspring produced shows rolling tongue. While when both the parents have non-rolling tongue, 34% of the offspring produced shows rolling tongue. Table 3 indicates the comparative data of rollers and non-rollers by different researchers based upon monozygotic and dizygotic twins. According to some researchers, when both the parents have rolling tongues, according to Matlock research, 18 of the monozygotic twins produced shows rolling tongue. According to Reddy et al., 43 of the monozygotic twins shows rolling tongue. According to Martin research, 15 of the monozygotic twins shows rolling tongue. According to Reddy et al. Dizygotic twins research, 81 of the dizygotic twins shows rolling tongue. When both the parents have non-rolling tongue, according to research of Matlock, 8 of the monozygotic twins shows rolling tongue. According to Reddy et al., 11 of the monozygotic twins shows rolling tongue, while according to the research of Martin, 5 of the monozygotic twins shows rolling tongue. According to the research of Reddy et al., dizygotic twins, 21 of the dizygotic twins shows rolling tongue. When one of the parents has rolling and the another has non-rolling tongue, according to the research of Matlock, 7 of the monozygotic twin shows rolling tongue. According to Reddy et al., 7 of the monozygotic twin shows rolling tongue. And according to the research of Martin, 8 of the monozygotic twins shows rolling tongue. According to the research of Reddy et al. Dizygotic twins, 30 of the dizygotic twins shows rolling tongue. The given graph is based upon the data that we have recently seen in discussion. This made it impossible to link the morphogenetic pattern of tongue rolling to the ABO blood group, while participants with blood group B and AB recorded an equal number of tongue rollers and non-rollers. Those with blood group A and O recorded a large distribution of tongue non-rollers. Conclusion. In conclusion, we can say that the null hypothesis is proved to be true, that there is no relation between the rolling of tongues and one blood group. Although the ABO and RH blood group system are among the most significant, they are inherited differently in population with various genotypes. Nevertheless, it does not play any role in the rolling of one's tongue. Here are some references. Thank you.
Thank you, Prachi. Thank you, all the presenters. Now, moving ahead with the review session, I would request Dr. Savitri Katlam, Assistant Professor, Department of Pharmacology, MGM Hospital, Kamote, Navi Mumbai, to briefly review the session on PPT presentation. Over to you, ma'am.
Am I audible?
Yes, ma'am. Yes, ma'am, you are audible.
Okay. A very good evening to one and all present over there. There were very diverse topics and some of them were very innovative. I congratulate all the participants, either they may be faculty or student. All of them presented very well. Thank you.
Thank you, ma'am. Thank you. Thank you so much for your valuable points and emphasizing the importance of research for the students. We will have a tea break and then we'll come back at 4:30 for valedictory function, where we will announce the best paper presentation. Thank you so much. We'll meet again at 4:30. Welcome back for a valedictory function of today's National E-Conference on Frontiers in Biological Sciences. Now I request our Honorable and Dynamic Principal, Dr. Giri, to present valedictory address. Over to you, sir.
Happy evening, all of you. Hope the day must be very hectic today, but every one of you must have got some sort of food for your brain. I know that there were deliberations on different aspects, especially in the field of zoology, biology, and every aspect which is related to biological science. There were presentations by the students and teachers. Thought-provoking papers were there, and really this particular conference could bring us together on the platform of knowledge. Dr. Savitri was there with us. She was assessing all the paper presenters, their contribution. Thank you, ma'am. You have put your all efforts in making this conference successful. There will be some prizes to research students as well as to the teachers. Some of you will get prize, all will not. So don't consider that you have not done anything, because prize is a criteria. It is some of the students are selected on the basis of one particular criteria, but everyone has touched that particular criteria. That is the achievement. And you consider this participation as one step you have taken ahead, that you have contributed and you will be the winner for tomorrow. With the positive approach, everybody will think and you will carry good memories of this particular conference of today. So once again, I thank all of you. Without taking too much time, I express my sincere thanks to all of you. Thank you very much.
Thank you so much, sir. Now it's the time for declaration of best paper presentation. I request Dr. Savitri Katlam to announce the best paper for today's session. Over to you, ma'am.
Yes. A very good evening to all. As sir has already said, all are winners, but still I have to choose the best paper. All have presented very nicely, and from different, they have touched the different topics. So I have to choose one winner in research teaching category and 3 winners in student category. So for the student category,
Ma'am, your voice is not, you have muted yourself. Yeah.
Now audible? Am I audible?
Yes, ma'am, you are audible.
Yeah. So first prize in students category goes to Alifia Bhaske for evaluation of phytochemicals and antioxidant activity of Musa acuminata floret extract. Second prize goes to Kashish Oja, study of bird biodiversity and mangroves in Navi Mumbai, India. Third goes to Vanchita, a survey on knowledge and awareness of anemia among female undergraduate students of Navi Mumbai. So these are the 3 prizes under student categories. Now, among the researcher, faculty or teaching staff, any guesses? all have done well, but the best paper goes to Mrs. Rashri Rajesh Sawant for characterization and antimicrobial activity of silver nanoparticles synthesized using scales of Indian salmon. It was a novel
Thank you,
research.
ma'am. Thank you very much.
I congratulate
Congratulations.
all the participants and the winners. Really, they have done good job.
Thank you, ma'am. Thank you so much.
Thank you. And I also want to thank the KLE College for giving me such a good opportunity as chairing the session. Thank you very much.
Thank you, ma'am. Thank you. Thank you so much. Thank you is such a prayer that cannot be seen or touched.
Thank you, ma'am.
It must be felt by her. Now it's time to propose a vote of thanks. I, Dr. Shashikala Prajapati from Zoology Department, take privilege to propose vote of thanks. Good evening to one and all the respected dignitaries, invites, participants, and dear students. I, Dr. Shashikala Prajapati, Assistant Professor, Department of Zoology, feel honored and privileged to get the opportunity to propose vote of thanks on the occasion of this national e-conference on behalf of KLE College, Kalamboli, Navi Mumbai, Maharashtra. Certainly, the national conference was very useful. We all got the opportunity to make aware ourselves with the innovative practices with respect to frontiers in biological sciences. Organizing a national conference is a challenging task. This cannot be possible without a great preparation, wise working hands, supports from every corner, and the blessings. On behalf of the organizing committee, I would like to express my deep sense of gratitude to our KLE management for the support in all possible manners to organize this conference. I am very much thankful to our principal, Dr. Giri, for his mentorship, encouragement, constant guidance, and support to shape the conference. Thank you, sir, for your support. Let me express my deep gratitude to our keynote speaker, Dr. Didi Jhala, Associate Professor, Department of Zoology, Biomedical Technology, Human Genetics, Wildlife Biology, University School of Science, Gujarat University, Ahmedabad, for taking out time from his busy schedule to be with us here today. I sincerely thank Dr. Didi Jhala sir for kindly accepting our invitation and sharing his vast experience and knowledge. I am also very much thankful to all advisory committee members, Dr. R.J. Verma, sir, ex-professor and head department of zoology, University of School of Sciences, Gujarat University, Ahmedabad, and Dr. R.V. Devkar, sir, assistant professor, Department of Zoology, Faculty of Science, the Maharaja University of Baroda. They supported in all possible manner to organize this conference. I sincerely thank to all the members of advisory committee for their valuable suggestions. I take this opportunity to thank our session chairperson for paper presentation, Dr. Savitri Katlam, Assistant Professor, Department of Pharmacology, MGM Hospital, Kamakhi, Navi Mumbai, for reviewing the session and providing her valuable comments towards the improvement of the quality of paper presented in the conference. I thank our technical team, Assistant Professor Sayma Natekar and her team for providing their support throughout the conference. I also thank all our authors who have submitted their valuable research to this conference. My thanks to the head of the Department of Zoology and convener of the conference, Associate Professor Dr. Usha Karunakaran, for shaping this conference so nicely and so timely. We have been lucky enough to be backed by a team of very motivated and dedicated organizing committee members. I cannot thank everyone enough for the involvement they have shown and the willingness they have expressed to take on the completion of the task beyond their comfort zone. I convey my sincere thanks to IQAC coordinator and my colleagues for their dedication and support. If time is money, then today you participants have spent millions for us. Thank you all for making this event successful. At the end, I bring sincerely apology to those who have directly and indirectly helped us, and I forgot unwillingly to take their names. Hope we all meet in the next event. Once again, thank you all for the support. Feedback link has been sent in the chat box in the YouTube also. Kindly fill the same so that you can get the certificates. All the participants are requested to rise for the national anthem. Thank you all. Yes.
सर्टिफिकेट्स कब तक मतलब मिल जाएंगे if
You will get it within 2 days. Within 2 days. Okay. You just fill the
मैम
paper.
जिन्होंने अटेंड किया है उनको भी मिलेंगे ना?
Yes, yes, yes. All
Thank you,
will
ma'am.
get participation certificate. Along with that, those who have presented paper, they will get certificate for participation also. Okay. And the best
Thank you,
paper
ma'am.
presenters will also get the certificates. Okay. Yeah, thank you all for attending this conference. With the permission of chair, I declare the end of this session. Thank you so much.